Morning, sir.
Good morning, sir. Dear friends, let me introduce our speaker. It is visible, Dr. Sir?
Yes.
Let me introduce our current speaker. He is Dr. KP Mahapatra, Principal Scientist, working in Division of Germplasm Evaluation in NBPGR, and he is a vast experience in NRM, Natural Resources Management. He is 2 decades of experience, especially in lower Himalayas, ravines of Yamuna, and humid climate and acidic soils of northeastern India. And he is a specialization in agroforestry and experience in ecosystem of northeastern India. He worked 11 years as extensive, and he led many extramural and internal projects also, more than 50 crores working, and quite intramural projects in the last 20 years. And he has a leadership in ICR Research Complex, Barapani. He given a successful leadership in division of crop production and agroforestry, and he established several gene bank initiatives in agroforestry items, and especially he worked as a PME cell in charge, and he is a head of the department in division of crop production and agroforestry in Barapani. And he introduced the first time in the world, we can say, that is IoTs in tree germplasm management, first time. And currently he is working as a principal scientist, and he is maintaining the field gene bank of agroforestry crops. And especially he introduced the geotagging facility and chip gene technology in our forestry. Geotagging facility will give us a very good and identification of easily we can do the tree specific locations. And he published more than 100 research papers, which are having more than 10 NAS rating papers also there, and 5 books and many book chapters, bulletins, and other things also. And sir, welcome you, sir.
Thank you.
And sir, being an expertise in your field, please kindly share your knowledge, and please express your knowledge and expertise on field gene bank management of trees crops especially. And thank you, sir. Welcome you, sir.
Thank you, Dr. Ravi. Good morning to all of you, all the participants. Let me share my screen first.
Yes, sir. It is visible, sir. Sir, please continue.
Is my screen visible to all of you?
Visible, sir.
My audio is also clear.
Yes, sir.
Okay. So, good morning, friends. I have been assigned the topic of management of tree genetic resources, as discussed by Dr. Ravi. We have started some work at NBPGR in the last 1.5 years. So the management of tree genetic resources, it's a broader topic, which by now you must have been covered with many topics on PGR management, most of which have been covered under the field crops. So tree genetic resources management will be dealing with how these tree genetic management sources are important, and how the things are going on the world, in the entire world. So in today's world, we are facing with 4 major issues. One is the population growth. The second one is the food demand, which is increasing day by day. Food, fodder, energy, and everything is increasing. land conversion is rampant, forest is shrinking, climate change is a certain reality now. So the future challenges are many and different also. Then habitat destruction is another issue. Like all the wetlands we are losing now, the wetland ecology is getting lost. So these are the main challenges the world is facing. So I have just tried to prepare my presentation in six broad outlines. One is the background, how things are there before us in the world, and the FGR movement, forest genetics resources, the history of forest genetic resources movement in the world. Then the biodiversity, the in situ and ex situ conservation. Few things you must have been told by now because it's the last day. So maybe many things have been told to you. But certainly I'll emphasize the importance of the tree germplasm and its relevance for conservation and how the world has achieved from its conservation. Then the field gene banks and its need, and a general guideline for field gene bank management, starting from its establishment to its information management and material distribution. Then how we manage at NBPGR, we have some experience in the last 1.5 years in managing the data of the tree germplasm, whatever we have in the gene bank. So as we discussed, we have the many challenges. Just a minute. So when we see the population, it is expected to be around 9.1 billion, and the food demand would increase by 70% than what we are producing now. And the energy and wood products, that is going to be more than 40% what we are producing now. When we value the agricultural production, it is around $2.6 trillion. And the population, that is food insecurity, even if we are producing more at present in India, but when we see the world scenario, around 860 million people, they are not getting enough food. The raw timber harvest, this is now at a very high level, 4 billion cubic meter. And the population depending on fuel load is 2 billion. And the population depending on natural medicine is around 4 billion. So looking at such a huge demand, which we have now and which we are expecting to get in future, maybe in 2050 or in 2070, for which the projections have been done. At the same time, the forest cover is shrinking by around 7 million hectares per year. And some reports are there where they have reported it is around 12 million hectares per year, the forest cover is shrinking. So as a result, the species threatened with extinction is around 32,000 plants, and the red list of threatened plants is around 20,000. CITES listing of tree species is around 900 trees. Many of you might be knowing that recently red sander has also been included, the Sterocarpus centellinus. Dalbergia latifolia has been included. Unfortunately, many people are confused with Dalbergia sisu. So the trade of sisu is also getting restricted wrongly or erroneously because of the inclusion of Dalbergia latifolia. So when we see these woody perennials, it is around 45% of the vascular plants, and the plants dependent on animals for pollination and other biological systems to continue is around 75%. So this is a huge figure for which I am giving you these figures because we will be discussing how these ecosystem and ecosystem services are essential for the tree genetic resources to evolve. So that was the purpose of showing you an idea that around 75% of our plants depend on some other animals for their reproductive biology to proceed. As we discussed climate change is certainly a reality. what we, if we recall our earlier reports, we are committed to keep the climate, the increase in temperature below 2 degree than what we got in the pre-industrial level. But that is not stopping. As the projections have been done, made that we may get as high as 4 degree higher temperature in future in 2070 or 2100. So if you see this report, the developing countries are demonized, that they are losing more biodiversity, they are doing more pollution and other things. But if you see the developed countries, they are already at a high. They might not be increasing, but they are already at a high in many of the parameters. So since the developing countries, they need more land to produce more, the population increase is high, the technological availability of better technology is less. So they are polluting more, but that pollution is because of the development. So if you see the global land use for food production, around 30% we have in the lands, out of which 70 is the habitable land. From this 71%, 46 is agriculture, 38 is forest. If you see this agricultural land, that is a typical misnomer that we need more land for agriculture, but we are using around 77% for livestock and only 23% in croplands. From this 23%, this is FAO data, 82% is plant-based food, and this 77%, it is providing only 18% of the calorie. So we are getting most of our calorie from here, even if we have area under crops is less. And unfortunately, this calorie, it is producing only 37% of the protein, and the food plants, the plant origin, is giving us around 63% of the protein. So you can see how this animal and plant things are going on. The area under animal system is more, but the protein and calorific contribution to the food basket is less. Then this is a, you can say, around 6,000 species we are primarily using. Out of it, around 1,200 we are using for food, 2,200 for fodder, timber and fuel and other wood-based materials we are using 1,000, 1,500 for medicines. Then these are the figures, many overlaps are there. These are the primary species we are using, but there are almost all species are being used by human being. But these are the species which you use for our major requirements. Then when we go to the history of FGR, when people started using the tree genetic resources or the forest genetic resources, because when we talk about the tree genetic resources, I'll be covering both the forest trees, which are used to primarily for timber and fuel wood, as well as the fruit trees. So the history goes back to 1787. The Germans in the whole world, Germans were the first to use tree introduction from different places. There might be many ancient historical things, like when Gupta period or Maurya period, how the trees have moved in a period of Ashoka also. But the recent history, when we say the modern history, we can say that in 1787, it is the recorded history of tree movement. The germplasm movement took place. It was initiated by the German scientists. They introduced some of the pines from North America based on the provenance information. Provenance, it might be a term new to you. Provenance, it is just a term synonymous to population, but it is used to more of the forest populations. Provenance, we can define it as a interbreeding population. It's a group of plants of one species which are breeding among themselves. So they are isolated. They are in some patches. You can think that as a provenance, because people started to realize that the seeds from different sources, from different provenances, they have different properties. So better provenances were giving them better yield and better products. Like many pine provenances, they were fast growing of same species, like Pinus sylvestris, Pinus dida, which were the uh, initial species which people started introducing. Then after that, around 1850, there were a lot of deforestation in Europe because Europe was developing fast. So it has come down to 20%. So the Europeans, mostly France and Germany, they started mass afforestation, for which they found shortage of good quality seed material. So UFRO, that International Union for Forestry Research, there is an organization called UFRO. They started massive provenance trials from 1907 to 1930. Mass scale provenance trials were conducted in many species around the world. Mostly they did those provenance trials in African countries, in entire Europe, America, and Canada. So the largest being in that period was 1100 provenances at one go. There was this trial was in pine that was conducted across Europe. So at that time, the major emphasis was Pinus sylvestris, Pinus dida, Abies alba, Picia abies, Laris decidua, and Quercus species. These are the 5-4 species which were primarily used for timber and pulpwood. So provenance trials took place during that period. But after the onset of the Second World War, it stopped. Again, it started in late 60s, and you can see the chain of species which were emphasized: Betula, Eucalyptus camaldulensis, Melina arborea, Pinus cassia, Tectona grandis, this is teak. Ajadi Rakta indica, this is a neem tree, Casuarina, and Parkia biglobosa. So this list is very short list. There are many more. Just I wanted to give you the major species which we at India are familiar with. Eucalyptus is quite common in the Nilgiri Hills and the southern India. It is believed that in India, Tipu Sultan introduced these species here. Melina arborea is Gamar. This is also a good timber tree. Pinus cassia, this is a pine which is available outside India as well as in Northeast. In Meghalaya, you might be knowing one species called Khasi pine. It's a fast growing pine. That is Pinus cassia. Tectona grandis is the king of timber, teak tree. It is known to everybody. Teak, in fact, it was introduced by the British from India to Africa. From Africa, it was again reintroduced to the Indonesia and other islands in the South Asia and Southeast Asia. Ajaridacta indica, it is the neem tree. We'll discuss how this neem exchange took place. It's Indian origin. Casuarina equisetifolia in the entire coastal belt of India, mostly the Coromandel coast. This is used as a coastal plantation. It's a fast-growing fuelwood species. It doesn't have much timber value. Parkia biglobosa. We have one species of Parkia which is very famous in India. That is Parkia roxburghii. I'll show you the species later on. If you see the introduction of tree species, Acacia mangium, it was it was introduced in 25 countries. India is one of the major country where Acacia mangium has been extensively cultivated. This is a good timber tree. We call it a substitute of teak. It's a fast growing. It may not be as good as teak, but fairly good for the furniture making. So we have quite a good population of Acacia mangium. Neem. Neem has been introduced in 91 countries. You can imagine how vast introductions of neem has been done. And interesting thing is that there is a alkaloid in neem that is called azadirachtin, that is obtained from the oil that is extracted from the kernels of the neem. The azadirachtin content of the Indian neem, the neem that we have in India, it varies from around 0.2 to 0.75%, maximum 1%. But the neem that was introduced from India to Africa, that azadirachtin content is around 3.5%, around 5% of what azadirachtin we are getting in the Indian neem. So we still do not know whether it is a G interaction or by chance some good neem material has gone to Africa and it has been naturalized there, and the azadirachtin content is more. Now there are efforts to, because now for the neem coated urea, we are importing neem oil from African countries, from Kenya, from Tanzania. Those are the countries where we are from where we are importing neem oil now to take forward the government's program of coating all the urea with neem to make it a slow release fertilizer. So now efforts are being made to bring back some of the germplasm from African countries to India and try it here. Unfortunately, because they are very long gestation period trees, so the trial may give you results after quite a long period. Casuarina acroscyphilia, because it is a windbreak tree and it is a fuel wood, fast growing, it has been introduced in 110 countries. Sorry. Eucalyptus in 91 countries. You can have teak. Teak has been introduced in 65 countries. So this is how the trees have been introduced from one place to the other. And some trees have been extensively distributed in many parts of the world. If you see the history of eucalyptus, eucalyptus has been introduced from a very small area of Australia to most part of the world. So you don't find much variation in the eucalyptus where it is introduced. There are many more introductions. If I recall, Moringa oleifera, Sahajan. Moringa oleifera was introduced from India to Cuba by Fidel Castro to fight anemia among its population, because moringa leaves are very rich in iron. So Fidel Castro introduced moringa from India to Cuba. Now every household in Cuba would be having a moringa tree. So you can imagine the impact of tree germplasm exchange on at the country level, at the continent level, and at the state level. If you see the Cryptomeria japonica from Japan, it has been introduced to the Darjeeling Hills, and it is performing much better in Darjeeling than its own original habitat. So there are a few more examples. This is just an idea which has been put before you. Before we go to some parts of our discussion, this is a slide just I wanted to show you how we define forest and agroforestry area, because many times we'll be discussing the data, how much area is under which system. So let us have some clarity on how the forest or agroforest area is defined. Because in India we have two terminologies, area under forest and trees outside forest, which we call TOF. So FAO is defining forest is any land spanning more than 0.5 hectare with trees higher than 5 meter and canopy cover more than 10% trees. So basically FAO considers 0.5 hectare land under tree is forest. But in India, Forest Survey of India, which estimates the area under forest, they consider at present 1 hectare as the area for which it considers it as a forest area. Because of the available technology in 1987, FSI was considering 400 area patch as one forest. Then the resolution, the spatial resolution increased because most of the things are done by remote sensing work. Because of the improvement of sensors, by 89 they have reduced it to 25 hectare. Now at present from 2001 onwards, they are using 1 hectare as the area which is considered as forest. But for agroforestry or the trees outside forest, to calculate it as a area under agroforestry, they are taking zero point 0.1 hectare as one unit. And when they convert it into forest cover, they convert it into a 70% equivalent canopy. It's not ground, equivalent canopy. Then we have two more definitions, which is a legal definition and ecological definition. Legal definition is the any area that is designated as forest by the government gazette, that is called forest, even if it doesn't have a single blade of grass. So that is the legal definition of forest in India. Whatever has been declared by government of India as forest, it is forest. Then there is a ecological definition. This is most tricky, critical definition, because by ecological definition, a forest is a forest when it has the woody perennials, it has all associated things in it. It has the animal, it has the microbes, it has everything, and they are interacting among themselves. Only then we call it as a forest. So by this strict definition, we do not have more than 10% area under forest in India. It is still less than that, where all the ecological processes are continuing. So since we will be using these terms in subsequent slides, I've just given you that idea. Now let us go straight to the biodiversity which we want to save. So the diversity, primarily, it must have been told to you that we have three types of diversity. One is the intraspecies, interspecies, and the ecosystem diversity. In agriculture, for breeding purposes, we focus quite a lot on the intraspecies diversity, which is nothing but a genetic diversity. Within the species, whatever genetic diversity is there, we measure it, we focus all our improvement programs, crop improvement programs on the interspecies diversity. Then we use nowadays quite a lot, we are using the interspecies diversity, just the wild relatives, crop wild relatives, and other things which some good genes are there which we want to integrate here. So primarily in crop breedings, we first focus quite a lot on these two types of diversity. But the ecosystem diversity or the habitat diversity, this is quite important in case of trees, because the evolution process must continue for the tree species to evolve. Just to focus it, I'll just show you a 5 minutes video, 4.5 or 5 minutes, which is Yellowstone National Park, how the the trophic cascades take place when the ecosystems are damaged, and how the ecosystem, the species diversity is affected, and why we must focus on this ecosystem diversity for in situ conservation, because many times we neglect this. But why it is important, there will be a 5 minutes video. I'll run it from here. This is, of course, a National Geographic website. Many of you might have seen it, but still those who haven't seen it, I'll just try to run it for them.
Sir, his video voice is there.
Yeah, voice is there.
Voice is not coming actually. Little bit.
It is not good to disturb now.
Okay. Then you can explain, sir. It's yes, sir. Even voice is slow, and you just
if it
explain.
is not audible to anybody, I'll just
Yes.
discuss it. I'll just speak it now.
Yes.
Actually, this is a video from Yellowstone National Park. The link is in there in the National Geography. It is called a trophic landscape. What happened? The Yellowstone National Park in 1950. If it is not audible to others, let me speak it. So what happens? The Yellowstone National Park, the wolves were there, which were hunted because they were a menace to people. So the wolf was wiped out from the ecosystem in 1920s. It was a lush green forest. It became a desert. So people were not able to know how a lush green forest could they did. They introduced again wolf Yellowstone National Park. So what happens? The first thing they did, they started bringing on the deers that are they are available in the Yellowstone National Park. So the deer population was reduced from 17,000 to 4,000 over a period of 10 years. So once the deer population came down, the vegetation around the rivers, they started coming. If you have seen the aspens and other things, this vegetation started coming up. Once this vegetation started coming up, so what happens once the vegetation started coming,
सर आपकी आवाज नहीं आ रही सर स्क्रीन आवाज नहीं सर आवाज भी नहीं आ रही और स्क्रीन भी सर नहीं आ रही है आपकी दो क्लिक आ रही है
Sir, okay. Sir, सर एक बार स्टॉप स्क्रीन करिए, स्टॉप शेयर करिए। हाँ, फिर स्क्रीन शेयर करिए। यस सर।
I think it has come now.
No sir, your image only camera yours is coming. शेयर स्क्रीन करिए सर। शेयर स्क्रीन। No sir, it's something. PPT is not visible sir, PPT. हाँ सर, अभी फुल स्क्रीन करिए। हाँ, पावर पॉइंट मोड। हाँ, ये वाला सर। हाँ यस सर, करिए। हाँ ओके सर, ओके। हाँ, आगे करिए सर स्लाइड्स ना।
अब हो गया, आवाज आ रही है ना? Am I audible now?
Audible
Yes,
now.
sir.
Audible.
You are audible, sir.
Audible,
Yes,
sir.
yes. My voice was unmuted by the other side.
Yes, sir.
Yeah.
Okay, sir. Visible now.
Now my voice is
Yes, sir.
also audible.
Now you can continue.
Yeah, thank you.
Yes.
So I was discussing about this. Could you hear me when I discussed about this Yellowstone National Park?
Yes, sir.
Participants, please.
No, sir. No, sir. We couldn't hear, sir.
But you could see the video.
Yes, sir. Yes, sir. We see.
Okay, this is a National Geographic video in the Yellowstone National Park in the United States. In 1920s, the wolves were removed from that national park, but because those were posing a threat to the human life. Unfortunately, over 50 years, that entire forest was converted into, it turned to a desert. That phenomenon is called trophic cascade, because the trophic level crashed because of removal of the wolves. So the forest got converted to a desert, and all the flora and fauna, they were wiped out naturally, because then in 1990s 100 wolves were introduced to that Yellowstone National Park. So when wolves were introduced, they started preying on the deers. The deer population was. Can you please unmute the participants? Yeah. So the deer population came to 4,000. Once the deer population came to 4,000, in the river side, the grasses started flourishing. And because of the wolves, the deers, they did not move up in the hills. So they reduced the destruction of the species or the trees or the vegetation on the hills started reducing. Means the vegetation was more and better. Once that better, the aspens and the pines that you saw there, they started coming up. Once they started coming up, the river started giving shelter to the beavers. Once the beavers came, the fish started coming. Then when the fish started coming, the otters started coming. So in only 10 to 12 years time, in 6 years, the improvement started to be seen. Within 10, 15 years, it became a full green, complete forest in 10, 15 years. So that the intention of showing is that there is a lot of emphasis on this ecosystem, because if you do not give emphasis to the habitat conservation for in the in situ conservations, the ecological processes or the genetic processes will not proceed. So by now we are doing all our efforts in interspecies. Maximum effort is on interspecies, a bit less on interspecies, and almost least on ecosystem conservation. You might have been told by Dr. Ahlawat in some of the earlier presentations when he presented about the wetland conservation in Majuli Island in Assam, where the Rufi frog and other things have been conserved. So the on-farm conservations are nowadays emphasized because it is required. And why it is required, that was the purpose of showing it, that we need all the organisms in the ecosystem intact so that the evolutionary process of each species can continue in its own way. If you don't do that, everything is freezed. That was the purpose of showing it. I'll try to be a bit fast because I have some other things to show you. So now the conservation approach lies in two things. One is the dynamic and static, because the genetic processes suggest whether it should be dynamic or it should be static. Because the dynamic process, we have the in situ conservations, the national parks, the sanctuaries, the biosphere reserves, the in situ seed orchards, then conservation provenances. Here, genetic process is conserved, means the process is continued. Then we have another conservation system, which is called Circa system conservation. We'll discuss in detail in the later slides. Then the XC2. XC2 is the seeds in bag. Most of the around 90% of the materials are conserved in the seed gene bank, in vitro gene bank, cryobank, field gene bank, and the botanical gardens. Botanical gardens are also an important ex situ conservation system. One data says per year around 500 million people, half a billion people visit botanical gardens across the world. But in this system, the genetic process is completely frozen, it is arrested. So why we do these conservations? The conservation, so let me bring in one more point. Whatever conservation efforts we do, the basic idea is always to capture maximum diversity per unit time, per unit cost, and per unit area. Isn't it? So, whatever efforts we take, we always revolve our efforts around this maximum diversity in minimum cost and minimum area and minimum time. Anyway, so these are a few things. What is the comparison of the static strategy and the evolution strategy? And this is the Indian scenario where we have this much area. You can say if you total it will be less than 10% area under the in situ conservation status. Unfortunately, in our country, this in situ is under the domain of Ministry of Forest and Environment, and ex situ is primarily under the domain of Ministry of Agriculture. Like NBPGR is doing all the ex situ conservation in gene banks, in field gene banks, and the in situ conservation is done by the MOEF, Ministry of Forest and Environment and Climate Change. So we have all these areas here. I won't go detail into it, but they are required. If I give you one example of this in situ conservation, in case of apple, in apple, there are some wild relatives of apple, pine, this Malus silvestri, Malus silvestris. All these are found in the forest of highland forest of Kazakhstan, Kyrgyzstan, and some parts of China, Uzbekistan. That Central Asia areas, you find the wild apple populations. Then maximum diversity is also available there. So that is the only place where you can conserve them. So, and of course, in Kyrgyzstan, some patch has been demarcated for conservation of wild habitats of apple. In India, if you have heard about the citrus gene sanctuary in Tura, Meghalaya, all the citrus in situ have been conserved. That effort was initiated by NBPGR long back. So the citrus gene sanctuary is there in Tura. Similarly, you have a coffee gene sanctuary in the Ethiopian forest. You have maximum diversity of coffee. So that has been conserved there. You don't have so much of diversity anywhere else. So these are the areas which have been demarcated. I don't have a slide to show you. We found some of the best materials of bale from the fringes of the forest. So still these in situ conservation, the national parks, wildlife sanctuaries, conservation reserves, all these protected areas, biosphere reserves are also there. They hold key to the conservation of many materials in situ. Now we can, I don't know whether you have been told about this circa system conservation. This is a conservation where we call it preservation of the plants or conservation of the plants or the farmer's field basically. Basically, when some trees were naturally occurring in certain ecological condition, there is massive land use change, but in the same eco-geographical system, isolated trees are there. In the farmer's field, in the backyard, in the home gardens, some trees are there. And these trees are excellent trees. You get a lot of good materials in the farmer's field also, so they are called circa system conservation. But they have their own limitations. What is the limitation? The functional diversity is most important. Because the plant you have there, it should have some relevance, like cultural relevance. I'll show you some slides. If I tell you in when I was going through the annual report, I found a good material of bale in the Basti area of Uttar Pradesh. So when I sent somebody to that area, I found that that was lost. Because when it was conserved earlier by the house owner, when they expanded and they constructed a new house, they removed it. So functional diversity is most important in case of Circacetum. Fine. Dasari mango. Many of you might be knowing that the mother plant of Dasari mango is one plant only, which is around 200 years old. Still, it is there in the Uttar Pradesh, Dasari village of Kakori, Uttar Pradesh. From that single plant, the Dasari variety has spread throughout India, and even it is found in many places in the world where it has been introduced from India. So these things can be conserved only in the farmer's field. This is another concept of Circacetum conservation. This is the polymoto type of conservation. This is Thai concept. It's a traditional knowledge of Thailand. Now it has been spread to, means it has been taken to the coconut germplasm conservation system and the World Crop Trust to many more places. I think it has been taken to this is somewhere in the Indonesian island where each variety is conserved in different islands. So what happens through that, a purity of the population is maintained. You can see the diversity from that area. And the certified seeds are produced from each separate island. But this is applicable only in those places where you have very isolated places. It's not possible to do in the areas where the continuity of land is there. In India, you have such areas in the sacred groves in Meghalaya and the northeast. There are those who have visited Shillong, they might have visited the sacred groves. The sacred groves, people conserve it because of the cultural significance. They think that their forefathers are living on those forests. So nobody is allowed to touch it. Then one is this, you can see this is called Parkia Rasburger. It is one of the most prized food in Manipur. This one pod, it can cost you as high as 20 rupees or even 50 rupees in some of their local festivals. There is a festival that is equivalent to Bhai Dooj of North India. It's called Nangol Thoba. So those festivals, this Parkia becomes very expensive. So people keep it in their backyard or in their fields, they don't remove it. So this is also a good example of Circacetum conservation. Then we'll come to the ex situ conservation, then the field gene banks. Let's say field gene banks, why do we need field gene banks? If you see the nature of the tree germplasm, they are very long-lived. Can you imagine how long-lived they can be? This is a plant, this is called Bristol cone pine in United States. It's Nevada. This is 4,900 years old, still a living tree. This has been published in Ecology in 1965. The girth is 21 feet, around 7 meters. You can imagine how big that plant must be. The trees are long living. Then they have evolved on a, you can see this. This is a Populus tremulidis in central Utah in America that is 80,000 years old. And one species, one tree is a one clone. If you see this, it's a single clone covering around 43.6 hectare area, weighing around 6,000 tons, is around 80,000 years old. The clones are dying, average 80 or 100 years, but the root system is 80,000 years old. So it is that big. So it goes through a lot of climate change scenarios, still it is surviving. Then you can see the size. This is a Sequoia sempervirens, which is around 115 meter tall and weighs around 530 tons. These are the peculiar things of the tree species. Then when we evaluate it at least 1/3 of the rotation for evaluation, for preliminary evaluation. For complete evaluation, we need at least half of the rotation, and the rotations of the trees are very high. In case of teak, in case of sal, it will be around 120 years. In case of teak, it can be 60 years. So all these things make it difficult for the trees to handle. Then I'll just skip it. The fields in banks, because I don't have much time now. These are the few things that are important. Just I'll tell you what is the economic benefit of the fields in banks. It can be millions. Improvements of crop due to CWR has been estimated to 115 billion annually. You can see and one evaluation was done on coffee. What is the worth of fields in bank for coffee? It is around 1.4 billion, because 3 traits were improved from the germplasm. One was the disease resistance to disease. Second one is the low caffeine or decaffeinated cultivar, because 10% of the global sale of coffee is decaffeinated coffee, and the high yield. These 3 traits were taken, and from the breeding program, the kind of benefit they got. So the value was calculated with a 5% discount rate over a period of 30 years is 1.4 billion. So that is the relevance of fields in banks, where we must start investing. If you see what you can do in case of field crops, it is easy to prepare the mapping populations, do the isolate the genes, and go to the gene level, and do all kinds of things. But in trees, the gestation period is very high. So from the germplasm, if you can do some kind of association mapping, the breeding cycle can be reduced drastically. So I'll give you one example. There are many. In case of apple, this has all been done from the secondary data. The work is not done by this group of authors. They have downloaded the available data of apple for blossom and shoot fire blight. These are the two economically damaging diseases of apple. And they did GBS. They just found around 55,000 SNPs. Did the association mapping. They found 23 QTLs associated with shoot blight and 38 QTLs associated with the blossom blight. And they could go for finding out the candidate genes for these two resistance gene to diseases also. So I'll not go to the epigenomic studies, but an interesting thing that it was done in, okay, let me show it also. In populace, even if they are clonally propagated because of the methylation and other processes, they found that 3 same clones in 3 different areas going through different phases of domestication, their response to drought was different, even if they are clonally same. So this is a classic paper that has come in PNAS. This can only be done in a field in bank if you have such collections and replicated to a good number of replications. Because in this case, it was more than 15 replications per clone. So in India, ICRAF, International Center for Research and Agroforestry, they do the conservation in field gene banks of most of the forestry species. They have 17,000 accessions of 81 different species distributed in 49 sites in 18 countries. So that is how ICRAF is conserving the materials in the field gene bank. And this is the distribution location where ICRAF gene banks are there. So this is all about what we discussed. And there is a broad guideline about field gene bank, which has been developed by FAO just recently in 2023. So this field gene bank, these processes are involved: acquisition by donation, acquisition. But this is the acquisition phase, quarantine, then curator has to curate it, then you have to field establish the field gene bank, then you have to manage it. So in the management, you have characterization and evaluation. Then regeneration and distribution is the essential part and the core part of any gene bank, may it be seed bean gene bank or the field gene bank. So I have given it in the writer, so I don't go details into it. But just to touch upon it, first thing is the site selection of a field gene bank. If you are to select, establish a new field gene bank, the species distribution models, Dr. Sembol might have taken some lectures on that. You can use them if you are not sure about the climatic conditions, how they are matching, because it will not exactly match. So you need to have some confidence level, 70%, 60% or 50% matching to select the trees which you want to put in your gene plan. Minimum risk of natural calamities. Lands do not have any history of nematode infestation. This is a big issue. Many times the entire crop is damaged because of nematode infestation. Buffer zone has to be there. It should be free from litigations as usual. These two, three things are important. Area for future expansion, because the trees are permanently on place, you cannot remove it. So you should have the sufficient area for expansion. Then vacant space for regeneration. After some time, you may have to regenerate some of the trees. If you have a patch of mango trees, you may have to regenerate it. So you need additional space for that. So always have some space for replicating your materials, because Dr. Thanabanan is there. I have seen in Hosur, in Moras, they want to regenerate some of the plants. So they need more area to replicate those plants from the mother tree. They have to replicate it, and then they can remove the mother trees. Then automation of the management system, this we'll discuss. Germplasm acquisition, there are standard rules. Of course, nowadays DOAs are there, because in tree species especially you have more complicated problems, because same accession will be having replications in clones, and it may be located in different gene banks, field gene banks, same clone in 5 gene banks. Then the DOI will help you. And most important is the phytosanitary standards. If you get some disease introduced into your field gene bank because of contaminated material, then the field gene bank is really at a risk of completely getting wiped out. This is the establishment, field management. It doesn't have much to do with the field management. The design, statistical design is ARBD. Spacing is more critical, because every species will require different kind of spacing. So when you plan it, it has to be planned much in advance, because you cannot have different species for different species and at the same time have mechanization. For movement of tractor, for irrigation, you need a uniform spacing also. So it is always better to plan it accordingly, so that if the lowest spacing is 3 meter, then the other spacing can be multiple of 3, like 6 meter, 9 meter, 12 meter. That will help you to mechanize and do the other stuffs in the field gene bank. Otherwise, if you have every species will have different species, then mechanization and irrigation, fertigation, they will be a big problem in all the gene banks. So we'll straight away come to the tree management system, data management. We have started in NBPGR just recently. We are establishing a field gene bank in NBPGR. This is a 30-acre plot. We have planned much in advance, allocated different blocks to different species. Then we have demarcated, given a block for all the exotic trees, because NBPGR imports many trees for distribution. So they need to be kept in one block. So we have dedicated one block for the exotic species, and we have prioritized spacing, and everything has been prioritized. Here we kept a 3 and multiple of 3 as the spacing. Around 205 accessions are there. We are still accusing, getting and collecting the materials. This is the bird's overview of the field gene bank that we have. If you just, I'm showing some trees are missing in between. If you see, we have around 59 NAG sites in the country where the field gene banks are there. Of course, all the field gene banks may not have the tree species, but few have. So when we manage the records of these field gene banks, we find difficulties of loss of data. These 4 issues are major issues for managing the identity of the germplasm in any field gene bank. Frequent loss of records. if like I joined NBPGR in the 2.5 years back or 3 years back. So when I joined, I had to start everything from scratch. I have to collect the records, talk to people, because the person who was managing, he left much before I joined. So certainly that continuity is lost. So when the continuity is lost, you start from the scratch, and many a times you wrongly identify an accession because of the missing plants that I have shown you in between. So field diagrams are many a times put you in problem. In some of the areas, the field diagrams are lost, the records are lost. So many things happen. So to counter that, we started a IoT-based, internet-based networking of the germplasm, where we have 4 things: a sensors, a gateway, server with web administration interface, and the mobile app. Basically, Internet of Things are that it's a network of computers and software where everybody talks to each other. This is a gene bank, this is a gene bank, this is a field gene bank, and they talk among each other through an automated system. So this is called a IoT-based networking. So we have 2 modules. One is that we use the radio frequency identification tags. Through a scanner, we tag this identity, then it is sent to the server. Now we are using a cloud server because we don't want to disturb the main data bank of the NBPGR. This is a pilot project. Once it is materialized, it is crystallized, then we'll replace this with a permanent server. Then we have a real-time monitoring through chips, where this monitoring system is a small, around 2.5 is 3-inch size. It looks much bigger here, but it is around 3-inch maximum, 2.5 or 3-inch. It has a lithium-ion battery in it. It continuously emits signal to a gateway. From the gateway, it goes to the cloud server. What happens? Any disturbance that happens to this tree, such a system, this similar system has been installed in Sandalwood in Bangalore also. But here the system is slightly different. So any disturbance to this tree, it will automatically send a signal to this gateway. From the gateway, it will come to server. From server, it will go to the curator or the person who are in charge of this through an SMS or through a mail. That something is happening to this tree. You just take care of this tree. So the data, when we scan it, we scan this data in this module. The common name, whether it is healthy, if some diseases are there or something else is there, this is fed manually when we scan it. In the app, these parameters, some fixed parameters are there. Some parameters like disease, healthy, these things have to be fed. Once this is fed, anywhere sitting in India, these are the data. These pictures can be uploaded, like we have uploaded the bale germplasm into the system. These are the 3 institutes where we have installed. And this is the dashboard. Sitting in your room, you can see how the trees are behaving, how many real-time sensors are there, how many things are there, and how many are in stress. 225 trees, when I took this screenshot, had some other problems, which it is showing. I think here it is showing that these are the trees which are under stress. So we can straight away start the troubleshooting program. We can talk to the person. So sitting here, I can check how the trees are behaving in Bangalore, IIHR, Indian Institute of Horticulture Research. Like it has given warning in 2 trees, initiate action immediately. So I need to talk to person sitting in Jhansi or in Bangalore, your such and such tree has some issues, take care of this. This is the investment and other things. And now we have started with Dr. R.N. Sahu will be taking the class, the drone-based phenotyping characterization of this tree germplasm in NBPGR. It's a big group. This person will be taking class next class. This person is Dr. Biradar from ICRA. So just by a drone, can we identify our germplasm, and we can automate it straight away from a drone to the server, the data can go. So this is a nutshell about what we have done in NBPGR. Thank you very much. Over to Ravi.
Thank you, sir. Sir, please on screen. Yes, sir.
Around 10 minutes.
Sir,
Yeah.
thank you very much, sir. Thank you very much. Actually, participants are requested. Kindly please ask any questions if you have doubts or queries.
Yeah, most welcome.
Sir, and you also can see in chat box.
There is no question actually. Good morning, good morning, good morning. Anyone, anybody who is handling, Dr. Thanavenan is there, if I am pronouncing properly, from Hosur. Is he online?
Central Circuit, from Central Circuit Board, sir.
Central Silk Board. Because they have a good plasm. For the audience, I would say, in India, if you have some finest...
There is a question.
Yeah,
Sir,
please, please.
Sir, for
Chat box.
how many seeds has to be collected for deposition of gene bank?
See, for seed gene bank,
yes, sir.
for seed gene bank, it is same, whatever you have been told about the crop plants. The 2000, 4000, and if it is a rare material, whatever you have, there is no limit to it. But when you collect the materials, I think Dr. Alawat might have discussed it. When we collect the materials, we collect populations. Is it? So that maximum diversity can be captured, because most of the trees are cross-pollinated, open-pollinated. So when we sample the trees, we sample from a batch, a provenance or a seed source. We bulk it. So when we bulk it, we take care, minimum 30 trees are bulked and the trees are deposited. When we find a good material, a unique material, then we collect seeds only from that tree. Then we deposit and we collect the buds for tissue culture or cryopreservation and for field in bank. So there we collect the clones, deposit for cryopreservation or tissue culture, whatever procedure is available or the protocol is available, and also in the field in bank. So this is all about population as well as individual trees. For seed numbers, it is same as you have it for the field crops.
Yes. And sir, and participants, if you have any doubts or other, sir, I hope there is no doubt, sir, from participants.
But just for information, I tell you that you have a very good field gene bank in IIHR Bangalore,
Yes.
in Lucknow Subtropical Horticulture Institute,
Yes,
and Hosur the Silk Board Institute. You
Yes,
have really nice field gene banks in those places.
Yes, sir. Sir, okay, then dear participants, if you have any doubts also, we are sharing Mahapatra sir number, contact email also.
My email also.
Email, and meanwhile you can post in chat box also, today and tomorrow also there. In bulletin also we are giving compendium. Okay, sir, let me, sir, sir, thank you very much, sir, for your nice presentation about forest genetic resources and the chip gene technology and how we can use for use the gene tagging, geotagging facility for tree species. And sir, thank you, sir. Thank you very much on behalf of organization team and other members, participants. Thank you very much, sir. And
thank you.
Okay,
so should I leave now?
Yes,
Bye-bye.
sir. Otherwise, you can continue also, sir, by
I'll see
and yes,
also.
Yes, sir. Yes. And dear participants, now it is a time for impact assessment, and Dr. Subhash will take the. Dr. Subhash. Yes.
Yes, I am there.
Yes. And it is a time for impact assessment.
I will share link.
Yes.
Wait for 1 second. into them.
It is most important and mandatory to fill the form. Yes, we can do the impact assessment of the participants. That is a pre-evaluation and post-evaluation.
Friends, I have shared a link
Hmm.
for a post impact assessment. Please go through that and do the needful. Yeah, you have 20-minute times. Please look at your chat box. I'm also posting this link in WhatsApp group also.
Dr. Sab, how much time you are giving?
20 minutes.
And please don't copy or don't anything. It is just for our impact assessment before pre and post evaluation.
Friends, only 25 question is there. So be quick because we have another lecture at 11:15. 11:15.
Yes, yes.
Sir, there is a problem. There is no tick mark to add option. Not adding the tick.
You are unable to do the tick.
Yes, sir. Yes, sir.
Wait.
Yes, sir.
Please, if you have any problems also, you can please mention in chat box or any.
It is not in edit mode.
It is not in edit mode. Okay.
No, not ticking there.
Dr. Ravi. Dr. Ravi.
Yes, I am in lawn.
You also do it.
Okay,
So
okay.
that we can know whether it is happening with everyone or some problem with them only.
No, it is going now.
But in my system, it's not there.
Okay. I think no, it is working.
I think it is problem with your computer or
Maybe
your
software update. Yes.
Yeah.
It is working. That
You can use that link sent in your WhatsApp group. Na? You use that instead of from this computer.
Okay, sir. Oh, okay. Okay.
You can. It is in mobile. It will applicable. Yes.
Yes, we can do this.
Please within fill the paper, not paper, fill the application form by 11 o'clock. Ankur, are you in online?
Ankur is not online.
Yes, sir.
How many applications received?
Till yet only 2.
2 only. Okay.
Yeah.
Yes. Dear participants, please kindly fill the mandatory forms. If you have any problem or query, please put in chat box or otherwise in group. Ankur, how many applications received so far?
The total 20 right now.
29. Okay.
20 only, 20, sir.
20. Okay.
Yeah.
Please, we have only 4 minutes time is left.
Friends, do it fast. Ankur, how many received? Ankur?
Sir, 29.
29 only.
Yeah.
Friends, do it fast. We have another lecture at 11:15.
Time is 11:10.
Good morning, Dr. Ravi.
Yes, sir. Sir, good morning, sir. Welcome, sir.
Okay. So is it a break right now?
Sir, just 5 minutes, sir. You can, sir, you can just check your slides also if you want.
That's perfectly fine. Yeah, I'll do that.
Just. Sir, it is.
Yeah. Saying something?
Sir, is it okay, sir? It is your slides. Sir.
Let me share with.
Yes, sir. Just we will check, sir. Actually, then.
I'll just share with you.
Yes.
Share the screen. Perfect.
Okay, sir. Okay. Sir, just, sir, okay, sir. Okay.
Fine.
Fine, sir. Fine. Sir, you
Okay.
please unshare your screen also. Actually, I want to introduce your this one now.
Still there is time, no? You have time.
Sir,
5 minutes.
2 minutes time is there, sir. 11:15 I will start, sir. Sure.
Perfectly fine. Perfectly
Yes.
fine.
Yes. And dear trainees, actually, I think a lot of hope. They submitted our impact assessment form. And meanwhile, if you have any problem or queries, you can post in our WhatsApp group.
So till yet only 40 applications were received.
So far, 40 applications received. And our next speaker also joined. We will start accurately at 11:15. If you have any technical difficulties also, you can mention in WhatsApp group. Okay, dear trainees. Meanwhile, you fill the application form, and let me introduce our next speaker. Sahu sir.
Yeah.
Yes, sir is in online. And now current, is it visible now? Yes. Yes. Our current speaker is Dr. R.N. Sahu, working as a principal scientist in the Division of Agriculture Physics at IIRA, New Delhi. And he is having more than 22 years vast experience in research, teaching, and capacity building in remote sensing and its applications in NRM. And mostly his specialization is hyperspectral remote sensing and radio to transfer modeling for soil and crop health monitoring, drone remote sensing, precision farming, plant phenomics, using the sensors at ground, air, and satellite platforms. And he is a leader in generating and mobilizing resources to lead different multidisciplinary and multi-institutional collaborative national and international remote sensing projects. And he generated nearly funding of about 1500 millions of INR. And he is having several awards or recognitions or citations. And his distinguished work, that is a Sahu sir, is UAV based remote sensing for soil and crop health, showcased to Honorable Prime Minister of India during 2016, 18 and 2022. And he is a member or expertise in several committees or groups or tasks or advanced working groups like DST on hyperspectral remote sensing. And he is a member to Science Campaign of ISRO and airborne hyperspectral remote sensing. And he is a member secretary of Task Force Group 1 on drone remote sensing of agriculture insurance of Niti Aayog, Government of India. And he developed a policy documents for drone-based crop-specific pesticide applications for Ministry of Agriculture and Farmer Welfare. And he is a technical member in a technical expert committee of DST and BIRAC DBT. And he is a visiting faculty to University of Nebraska, USA for remote sensing-based agriculture drought monitoring. And he is besides all these things, and he is an expertise, and he is a best teacher. He guided 8 PhD students and 2 MSc as a chairman, and more than 40 he is a co-chairman and members. And he published more than 240 research papers, 240 research things, and 120 research papers in high-rated journals and other documents and trainings. And dear friends, you know, this is a time for unarmed vehicles, UAVs or drone-based technologies. And nowadays we are widely using in different functions or any other things. We are widely using a drone technology. And now, sir, it is my pleasure and honor to me. I am welcoming you, sir, on behalf of team and other members, organizing team and other members. Please kindly share your experience and knowledge to our participants, sir. Welcome, sir. Welcome.
Thank you, Dr. Ravi.
Yes.
And thanks to the organizer giving this opportunity to share some of our experience on this technology for high throughput field phenotyping. Before I start, I just would like to see whether my slides are visible.
Yes, sir. Visible, sir.
Okay. Just I want to hide this one so that it doesn't. Okay.
Yes.
So today's my deliberation would be on drone remote sensing for high throughput field phenomics. I think all you are expert on your domain of field phenomics and genomics, and also I am totally novice for this field, but I would like to talk a little bit on how the sensors and drone can be used for this purpose. And you can correct me if at all some positions, some places, if I'm wrong in saying some of those interpretations. But it is always, I'm happy to how physics can interact with the plant science team to see, interestingly, to look for not only phenomics and also genomics. Recently, we came out with a wonderful results. I'm not going to say here because that is nothing related to drone remote sensing. So QTL mapping using fluorescence imaging, also hyperspectral remote sensing, we could do that. And soon we are going to publish it in a Journal of Experimental Botany. So what I mean to say here is this is what the linkage we can always develop with a multiple team to come out with the target problem. So why at all we look at the phenomics or precision agriculture? I think this is what the slides talk all about that. The enhancing crop production can be approached by majorly two things. By is how you can increase the genetical potential for the yield. That's by the smart crop or the phenomics, genomics, what you people have been working since long, and you are expert on those lines. So that is what the phenomics aspect, where if we can meet the yield gap, what we have with respect to potential and attainable and actual yield. The other aspect is if at all you have a crops and how you can manage the nutrient or other resources using site-specific input management by precision agriculture or prescription agriculture, what are smart agriculture, and how the sensors, data analytics can play a big role for that purpose. So this is what today we are going to talk on that only, on the field phenomics. So this is what the topic today. We are going to talk on more onto the phenomics than the precision farming. So coming back to the drone remote sensing, since we are going to talk up, so let me tell a little bit about what do you mean by remote sensing. Is nothing but how you can retrieve the information of an object without being in physical contact with that. That's what the remote sensing, and what you have been doing right now is nothing but remote sensing. You are using your biological sensors, that's 2 eyes, and which is working in electromagnetic spectrum of 400 to 700 nanometer. And you are using the light source as the universal energy, which is reflecting on the screen and going back. And you are able to assimilate on your central processing unit, that's your brain, and able to interpret what the information is. So this is all together is remote sensing. Why at all we are looking for remote sensing is to look beyond visible eye. That's what here we are looking at. This is what the visible region here. So if at all we can go beyond, like you look at the X-rays, then you can see what is the problem on my bone. Or if you look at MRI, you look at PET scanning, you look at any other instruments which you can't see in the visible range, you'll be able to retrieve some information in those ranges. And that's what to make you more rich with the information content. That's the purpose of having remote sensing. So remote sensing is to look beyond the range which we are not able to capture or retrieve the information, so that we can get much better information and go for the better decision making. So if you look at the, so that's why remote sensing started something around from 400 to something around 3000 nanometer. That's what optical remote sensing. Then beyond that, that's called thermal remote sensing. Then beyond that, that's called the micro remote sensing. So most of the sensors, what you can see all around us is 95% are on optical remote sensing, which works from 400 to 2500 or 3000 nanometer. And beyond that, there is the thermal remote sensing, like you have a thermometer, you have a thermal imaging camera. So that's what the thermal remote sensing. And beyond that, you have microwave sensors, where which works beyond the thermal range, and penetration power is better, where you have a country like India, where you have a lot of bad weather, but still penetration of the energy will be better, so that you can get a wonderful signatures information about the crop or soil or whatever the target you are looking at, in spite of the bad weather. So that's what the advantage of having microwave remote sensing. Then coming back to the technologies, like we have black and white, one single channel will be there, that's called panchromatic. Like you have a thermal imager, which will be simply a single channel. But if you have a RGB camera, like in a mobile, that is a multispectral, you have a 3 bands there. So you are able to see wonderful color photographs, and you are able to discriminate which is having what kind of color with respect to the target features. So that's what the multispectral is. That is more than 3 or 4 or 5 or 6 bands what you can have. So that's what most of the satellites have, multispectral sensors to characterize the target what you have. And we are talking more related to vegetation right now. So most of those bands, for example, you can see 1, 2, 3, 4, 5, 7. These are the multispectral sensors which is available, and you can characterize the plant very nicely. But if you look at beyond multispectral, that is called hyperspectral. That's what you are able to see very minute, every 1 nanometer interval, you are able to capture the data, and you are able to see the pattern of the signature. Like, for example, you see the green one, that is the vegetations. So at every 1 nanometer, you are able to capture the data, and you are able to see a lot of up and down in this signature. That means the energy interaction with the target with respect to its biophysical, biochemical properties is going to be unique with respect to those spectral ranges. And because of that uniqueness, you are able to see or characterize the parameter. For example, look at the chlorophyll content. That's the observation which takes place here because of the photosynthesis. Here there is a lot of internal reflection takes place. So in near infrared, it goes back, very high reflection you are getting. So, and that all depends on the cell arrangements, parenchyma cells, vocals, whatever you have. And then down, you have down here, the drop down in 2 places. That's because of water content. So this is a very 2-3 parameters I'm talking. But when you see starting from 400 to 2500, there are many biochemical parameters which is responsible for this kind of pattern to see. Unlike the vegetation spectra, when you see the soil, you see more of a uniform. So that's what the challenge what we have for the soil. But for vegetation, it's a wonderful interaction. You can see always, you can characterize much better than any other target. So when you see the multispectral and versus hyperspectral, these are the few bands what you have in case of multispectral. As I see in case of RGB, you have only 3 bands. You will be able to get information only those 3 bands. For example, you have something around 6 bands here. You are able to get only dot points in those 6 bands. So whatever the responsible parameter you can see in those bands only you can characterize, not beyond that. But when you have a hyperspectral, you have a continuous spectra. So each spectral part is corresponds to something on the ground on the parameter. For example, you can see in that black table, you can see the kind of parameter which you can look at. What are the bands you have? This is just a few parameter I'm given as example. You have all kind of parameter you can see starting from something around 400 to 2500. You can characterize them. So name it water, lignin, cellulose, protein, nitrogen, or chlorophyll or anything. You can characterize much better way than what you have in the panchromatic or multispectral. So that's what the beauty of having this kind of sensors. So just to give an overview how the capability we have over the time with respect to technological advancements. So when you put those sensors on the ground, like you have with your hand, or you have a mobile platform like a tractor, or on a standing still platform, that's what the ground remote sensing we call it. And when you put on the airborne, that is what unmanned aerial vehicle or manned aerial vehicle. So that is called airborne remote sensing. And when you put on the satellite, that's called satellite remote sensing. So today we are going to deal with the airborne part. So we are skipping ground and satellite for the time being. Whenever next opportunity, we discuss all those things together also. So how we can capture those variabilities from the lab conditions to the field conditions, and how that variability can be captured. So drone is nothing but in between all these ground and also satellite remote sensing. This is what the drone is. So drone is, and the UAV is nothing but unmanned aerial vehicle. And drone, you know, the name came from a honeybee. The sound, the drone is otherwise called the queen honeybee is called the drone. So that is, drone is nothing but dynamically, remotely operated navigation equipment. That's what the drone stands for. If you expand, if you go to Google, you can see that. So that is what the drone, we call it a keyword as a drone, but the expansion is this. So this is, and again, when you look at the synonymous, there's nothing but the queen honeybee, which has a signal passing on to his bees to collect the data. So the drone name derived from there. So nowadays it's very common. And this drone is nothing but the airborne platform where you can fly on an altitude as you like to. And again, it can sandwich between the ground and also satellite. So when you move from a ground, a single pixel, to the satellite is something like an ecosystem, bigger ecosystem you can look at, whereas in the ground, you have a single pixel or a few patches where you can get the data. So that's what the capability what we have over the time now. So today we are going to talk on the drone remote sensing. So when you talk of the drone sensors, so I call it as a CAD scanner for a crop or soil health monitoring for either a high throughput field phenotyping or precision agriculture. For example, I have given here IRE field with the wheat crop standing with the differential treatment of water, normal conditions and nitrogen stress. You are able to see the color photograph, how differentiated well, and you can, layman voice, you can see how the different treatment has been captured very nicely, not only in the field level, also in each field also, if there are 7 to 8 or 10 genotypes, how they are different with respect to that. So you'll be able to get all those details more in the subsequent slides. So the drone remote sensing is nothing but a CAD scanner on a lower air platform for your field or any target you are looking at. So if you talk of drone technology nowadays, people are very common now drone in ministry, and also many places, many startups have come out. So if you classify them with respect to their applications, so drone technology first is the surveillance, is the mapping, discrimination, the crop health monitoring. I'm just going to confine to agriculture only. I'm forgetting rest. It has a huge applications beyond agriculture also. But when you talk of agriculture, it's nothing but the mapping and discrimination of crops, mapping, monitoring of the crop health. That health may be the biotic or abiotic. So biotic, pest and disease, water, nutrient or heat. Then also crop growth, yield estimation and damage assessment. And again, high throughput field phenotyping for breeding and also soil fertility. When you come back to the drone as a input applications, most of the time it is used nowadays, very common. You must have heard the word Kishan drone. What Agri Kishan project has come up, and I think NBPGR and many other ICI research institutes got some funding resources to buy drone. So that is purpose is to how we can make it popular among the farming community and also in agriculture. So that's mostly for input applications for pesticides, herbicides, or liquid fertilizer. And all this possible provided you have a variable map created by drone or satellite remote sensing, where what kind of health condition is and how the variation is. Accordingly, we can go for the input applications. So drone as a variable rate technology is for that precursor is the drone-based remote sensing or crop health monitoring. Then also drone is used mostly for the bombing and also broadcasting and also many more purposes. So to capture that variability with respect to the soil, with respect to the crop and environment, and finally optimizing the yield, that's the purpose, and how drone can be helpful for that purpose. That's what the research right now going on. So today we'll confine tomorrow into the phenotyping, high throughput phenotyping, how we can do it. So coming back to the technology, the sensors, what is available right now. So there are many different sensors with the different resolutions of RGB camera. Most of the drones you can see is with the very low cost RGB camera with having very high megapixel capability. And also we have thermal sensors with the different resolutions and also there are different multispectral sensors nowadays is available. That's called Mica Sense with Red Edge, Parrot Sequo, Altum. And recently also DJI came out. DJI is a known brand of China, which is manufacturing many drones. And they have also come out with some multispectral camera. And recently there are some modification on this multispectral camera with more precision, more high resolutions. Then coming back to the hyperspectral, again, when you go beyond multispectral, as I told you, 4 or 5 bands, there are sensors which is available, which is hyperspectral, but a bit expensive. And there are certain parameters, traits, which you can't capture in case of multispectral, you will be able to see in hyperspectral. Then coming back to the LiDAR, again, LiDAR gives a more detailed information with respect to the plant surface, height measurements, and also morphometric parameter retrieval. And that can be used for different purpose of the crop phenology or crop parameter retrieval. Then the last one, which is almost now recent innovations, came out with is a microwave radiometer. It's a microwaves, like you have microwave sensors on the satellite. Similar sensors are also nowadays available for the airborne platform. And that's what we should be able to see how best we can use these sensors. Even if you have a bad weather, you can characterize different parameters, which is not possible using this optical or thermal remote sensing. So these are the all possible sensors which can be used on the drone for plant phenomics or high throughput field phenotyping. As at the beginning also, I told you, in the satellite cannot be used every time as you like to, or the resolutions you like to, or the small field experiment you do, or the kind of sensors you look at, because once satellite is launched, everything is fixed. But in case of drone, it's very, very flexible. It's a platform, it's a board, it's a robot, air robot, where you can fly as you like to, at what height you want to fly. Based on that, you can decide your resolutions, what sensor you want to fly. You can plug in, plug out with respect to RGB or thermal or multispectral, hyperspectral, whatever, and the time when you want to fly. Today, just right now, there's a bad weather outside. For example, it is hazy. Maybe after a few hours, a few minutes, you can see the bright sunshine. Then you can go out and capture the data. So that's what the flexibility lies with. That's what the wonderful opportunity what lies with the drone remote sensing. Then again, most important is the real-time data availability. Once you fly the drone, you can capture that, you can get the data within no time. There itself, you can download the data, or you can transmit to your lab to see what is happening in the field. And it's a long run, it's a low data cost. Once you invest something around 20, 30 or 40 or even 100 lakhs, so it will continue for years to come, and you can analyze the data. The kind of expenditure you incur otherwise is going to be very, very cheaper when you look at the drone technology. And you have a high spatial and spectral resolutions, and also temporal resolutions as you fly as you like to, and also the kind of sensors you are going to use. Again, there are techniques, there are pipelines, methodology, other softwares available which can made huge to process this data and make it very simpler analytical format, very simpler format anybody can use for. For example, you can see how the drone flies is a kind of the scanning it does and goes with like this is a field of view angle, what are the swath it covers. Then sensor has its own resolution. That's what the IOV instance of field of view or the pixel resolutions you can scan through. And everything can be done by two approach. One is either manual mode or the automatic mode, and that can be done. You can see the automatic mode is automation is done like you can plan where to take off and how to capture the data. This is somewhere in Cuttack, a small field where we are capturing. Beyond that, there is a pond. So you can see the kind of a mission planning has been done. So it will start from here, go down here, capture the data, and come back, and everything you have to decide before that. So you can have the kind of overlapping, like you stitch a cloth, keeping a overlapping, so that so same also the images can be stitched together to make a bigger image. Orthomosaicing, pre-processing can be done. So that overlapping 70 or 80 or 60%, that has to be given before you take up flight, and at what height you are flying and how you are flying. That all decides what is the technology you have, what is the drone you have. So there are two parameters what you mostly decide is, in case of drone, if at all you need to go for procurement. One is what is the payload it can carry, how much weight it can carry, whether it can carry 500 grams, whether it can carry 1 kg, or whether it can carry 10 kg. So that decides also your cost. That decides also what the capability you have with. For example, your drone can carry only 500 grams or 200 grams. That means only one sensor, like RGB sensor or few multispectral sensor, which weighs something around 50 grams to 100 grams or 250 grams, that can be taken up. But there are sensors which is very heavy weight, like you have a hyperspectral, which is, of course, the one which we have is around 600 grams. But there are sensors which weighs more than 1 kg or 2 kg, putting a gimbal there and all those things. So it all depends the capability, the payload, what you can take up. One. Second is the endurance time. How much time it is going to fly? What is the battery power you have so that it can fly for 10 minutes or 20 minutes or 30 minutes? So that decides how much area you are going to cover up. So for example, you fly at 20 to 25 or 30 meter height and looking for a area like a 5 acre. So 20 meter to 30 meter and a 5 acre, it takes maximum something around 19 minutes if at all you fly a multispectral camera. So, but when you fly with a higher height, then it may take much lesser and bigger area. But again, resolution is going to be less. So to decide at what height should I fly, at what time I should fly. So those are the SOPs, standard protocols, which we have published. And I think that is available also in HCP website from IRI, which you can download and see, starting from procurement of drone to integrate the sensors and calibrating the sensors to fly. Everything is available for that purpose. So I think I should skip this slide. This is a movie file which last time we have flown the hyperspectral camera and sensor viewing. So this is a drone which is flying at a height at something around 20 to 30 meters height and capturing the data. So let me stop it here. And this is a drone which we started sometime in 2016 before Prime Minister visit. So that's what our joining started in IRI, coming back to when you look for the drone technology. But when you look at 1969, fast airborne remote sensing was happening in IRI in Kerala, where coconut wilt disease was detected. That was nothing but a manned aerial vehicle where the sensors were there, and we could see that. So that's what the story started with IRI, something around the 1970s, the 69. And now, of course, that was not the drone, that was a manned aerial vehicle borrowed from Russia. Camera was borrowed from US, and ISRO, IRI, and NASA working together to detect coconut wilt disease. So you can see the own-made drones, which was costing something around 20 to 30,000 that time. We tried to throw in our Phenomex field in IRI, sometimes in 2016. So this is what all about. Coming back to the sensors, just a little story so that quickly we can understand. So this is what the radiations you receive when you look at the plant and put a sensor. You can see the kind of a spectral, the radiance, the energy what you receive. Why I'm showing this is a slide which was borrowed from a colleague from NASA called Robert Green from JPL lab. So it says a basic process of photosynthesis can be captured by the spectroscopy, by the sensor what you have. So you can go beyond simply qualitative analysis and go for quantitative analysis and also understanding the all physiological processes of the plant, having this kind of sensor capability. So that's what the beauty of the sensors what you have. Look at the LHS and RHS. So left-hand side, you have water, you have a carbon dioxide and the energy. Everything can be captured because each part of the spectrum is responsible for the component you are looking at, both left-hand side and right-hand side. So you can make a process understanding what is happening and how much water is got trapped, how much carbon dioxide is required, or how much is the carbohydrate produced, or how much oxygen is produced. So this is what the beauty of having the sensor, the science to relate them. So this is what one example is given. So like that, you can, if at all you have a census like this, you can characterize the plant with respect to all the parameters you are looking at. So keeping that in view, so this is what the facility recently we have and the platforms. So these are some of the boards or the drones what we have with the multispectral, with the RGB sensors, with the multispectral sensors. You can see some of those sensors what we have. So this is a Parrot Shikwa, which has only 4 bands. This is again a multispectral sensor called MicaSense Red Edge. This is called MicaSense Red Edge P because you MicaSense Red Edge with the thermal camera. This is called Altum, where you have 2 dots. You can see here, these are the thermal camera, and this is a hyperspectral sensor. So most of the drone remote sensing for agriculture, field phenomics, you can see people have been using all these sensors. Of course, hyperspectral, very few labs they have in the world. So these are the sensors which is available. But recently there are little changes with the sensors. If anybody is very keen to work on that and procure or go for the science, always you can contact us to help you out. So, and this is what I was talking of SOPs, manual and drone remote sensing, which we have developed. This is a kind of software which we try to develop for the ground non-imaging sensors, how you can do analysis. That's called HyperAgri. We recently also developed a drone agri software, which whatever the drone data you capture, how you can analyze, come back. I think next slide is going to talk on that. And this is what a very small drone lab we started with a very honest beginning to see some of those sensors with the drones we try to do in the IRE field itself. And of course, we have a big data analytics lab. All these are developed under an HCP program, and we try to see how best we can use for the plant genomics and plant phenomics program. We have an NHCP-funded plant genomics for the crop improvement. Under that, how high throughput field phenotyping can be done. So we use Nanaji Deshmukh Plant Phenomics Center. We use our experimental field using drones and also mobile platform. So, so this is what the phenomics. I think other day Dr. Vishwanathan might have taken more detailed deliberations on the plant phenomics. Just quickly to go through is here. We are using drone in the field phenomics. What happens is the plant is still, plant is not moving anywhere. Only the sensor is moving all possible directions as you like to. And on the drone itself, the kind of sensors we try to use is RGB. We use a hyperspectral, we use thermal, and also we use multispectral sensors, as I said you. So this is what we do in the field. And also we have a mobile platform. So this platform is having something around 1200 feet boom. And the kind of sensors what you use in the drone in the phenomics center, same sensors also we try to integrate over here and move in the field with a GPS guided so that automatically data is captured. And this is again a facility which was there. Again, is a frame where sensor moves on all possible directions on this frame and crop is grown over here. And in case of phenomics center, the sensor is fixed, plant comes on its way automatically and data is captured. So we try to see how best we can do high throughput phenotyping, starting from a controlled conditions in the 25 kg pot with RFID code and also climate controlled greenhouses to the field conditions where you can see a lot of variability with respect to not only the genotypes, but also the environmental variability, soil variability, managed variability. Because that is the key issue remains when you go from the lab conditions, which is the controlled conditions having 25 kg soil, when you go to the field, it's an unlimited environment available for the plant to interact, go down the route, seepage losses, many practical challenges what we have. So whatever the methodology we develop in the field lab conditions, how we can go to the field conditions. So this is what the phenomics center. Until now, we have created something around 16 million images to capture and to go for data analytics and developing. I think I can skip the movie file also. I think Dr. Vishwanathan might have shown you the movie files. So, so this is what the facility, I think, the kind of data we generate. This is with, I think this is second to the largest facility in the world, having 1200 carrying capacity of the ports, and also the kind of imaging sensors what we have here. So coming back to the data analytics, this is what the data analytics we try to develop with respect to the data what we generate. And this is what the drone agri software what we in-house we try to develop. So this is for the two purpose we try to do, how drone data can be used, whatever data is captured in case of field experiments, like we design the experiment with respect to breeding, altogether is going to be different. What you do with respect to precision, altogether different. And what you do in the farmer's field is going to be different. That's the reason why we have two windows for field crop experiment with a layout, and also like you have 3 lines of one variety, like you have 100 varieties growing in one treatment. So that kind of design can be done. For example, you can look at here what happens. So this is what the differential treatment you have. Again, within each plot, there are around 5 to 6 genotype you have grown, and each genotype of 3 lines or 5 lines you are growing with. So this is what the kind of a experimental design we have made it, and in the software itself we have incorporated, so that you can analyze, come out with a thematic product like this, and you can generate all possible. For example, you can see here, enter the number of rows, 9, and the number of columns here, and within that, how many number of lines per genotypes you are growing with. Whether you want to do it horizontally or vertically with respect to field, you can do, and the kind of sensors you are going to use. All the listed sensors are here. The kind of indices you want to calculate, you don't need to use your own knowledge or anything. So all the indices are listed over here. You click on the sensor, automatically those indices, which is possible through that sensor, you can do. Also, you can develop your own sensors, own indices. And also we are trying to see how process-based modeling can be done without these indices. You can go for the mapping. So this is what a visualizations, mapping, and also heat map generation, everything can be done automatically. This is what in-house program, what we try to develop it. So coming back to some of those experience what we had with. So this is what the IRE field we try to grow with rice and wheat. That's what the experiment we had in the plant genomics with a NSF funded program, very ambitious program what we had. So under that, we try to see how we can capture the data. For example, this is what the field we capture the data with a mission planning. And this is what the data, what you get captured, all the red dots you are able to see. These are all the frame-based the data is captured. If the sensor is tilted a little bit up and down, you can see how the image looks like. So those part is not taken into consideration for processing the data. So it generates all the cloud points, and finally you get a wonderful image generated out of it. So this is what the camera we try to use for that purpose. And this is what the stitched orthomosaic image, what has been created, and this is what the different experimental field. This is what the phenomics field what we have with the plant physiology, physics, and plant the genetics division. We work together. This is what the rice field with sometimes in October, some of the fields already early harvest has been done. And this is what the experimental field with physics with the different experiments. These are some other fields also. So this is what you can see and wrap over the Google Earth to see how wonderful it looks like. And this is what, if you see that you capture that data, what I was saying. So this is what the data you are able to see. This is what the differential treatment given with respect to drought and nitrogen. And you can see the layout which was there with respect to the growing and the genotypes which was there. And these are the indices which was calculated, called normalized differential range index, which tells about the nitrogen response. And you are able to see the differential response in the field conditions from not only from the treatment to treatment, and also within the treatment, the genotypic response you are able to capture. Very wonderfully, you are able to see is the low values with respect to drought and N120, D and N0. And when you go for the irrigated and N120, your crop health looks to be much, much better. And again, there is a differential response from the genotypes. So that's what you are able to see here. The same thing, what I was showing here. And also with the time you can capture those data, you can see the same NDVI image in the 80 days after sowing, 160 days after sowing, and 120 days after sowing, how the differential response. Again, you can put them in a heat map to see all these 29 genotypes, how they're responding, how they're ranking with respect to the differential treatment given on the different dates. For example, 9th February, and you have 7th March, you have 21st March. So this is what these are all automation done by that drone agri software. You can rank them, you can rank the fields with respect to the variability on the genotype and also field level. So same is for the precision agriculture, of course, we can do. So each field can be ranked 1, 2, 3, 4 like this, and you can rank them with respect to the response what you get with respect to the treatment. Same also we are trying to do with the chlorophyll mapping, how you can do simply using whatever the ground major chlorophyll you have, and also go for the prediction model and derive this data. And this is what I was talking to you, the rice field experiment, where some of the fields was early harvesting has been done. So you have the differential treatment of drought, control, and nitrogen. And you can see the yield predictions which has been done, or biomass prediction which has been done, simply using a low-cost sensor of RGB. And you can see the mapping, the color itself shows how the variations with respect to the biomass and what are those response with respect to treatment, drought, control, and nitrogen, and also within that, how the genotype is. So what I mean to say is, this is what the quickly, non-invasive, rapid estimations you can do, provided your prediction model is really good. So how good that prediction model all depends what approach you are going to follow, whether it is a simply empirical approach, a regressive approach, or machine learning or deep learning approach, or process-based model. So till now, what I am talking to you is all the empirical models what we have developed. For example, you can see here, this is AX squared plus BX plus C. This is simply empirical regression model which has been developed, and you can see the wonderful biomass predictions. Slowly, we'll go to the machine learning and finally process-based modeling. So this is what the thermal image. Again, these are the differential experiment which has been done. So there are many experiments with respect to ERI germplasm, and there are some experiments with respect to recombinant inbreed lines of 185 sets. And these are all different experiments what we had. So we try to capture thermal image using this camera called Altum, where you have two thermal sensors. So the same time you have a multispectral, same time you have a thermal, and you will be able to capture this data. And so you are able to see the temperature distribution. And this is what the heat map generated for all those experiments which has been created with respect to real cell genotypes what you have. So this is what the differential response always you can capture and map them together. And that is very rapid. Once your protocol is established, it doesn't take time to do this. So this is what live predictions, which you could do in some experiments and the different dates you are able to do. Again, this is all regressive models. Coming back to the water content, this is an elevated platform, of course, not on the drone. This is a hyperspectral sensor. We try to capture the data for the wheat field, and you are able to see the different indices, how medium and low, and what are the stress conditions where stress is very high, where is the stress is very poor. This is something around 139 genotypes grown in the field, and capturing differential response with respect to the water stress. Then this is again a 10,000 land races. This is again I think NBPGR is involved in this program. So this is a genetics field of our respected directors program with Gopal Krishnan involved here. So 10,000 land races, which is available in the country, which can be captured. We could capture the data in 10 minutes. And this is what the kind of process you can see. Let me go through very quickly to simulate a 3D image, a simulated 3D image, and making a walkthrough in the field can get you a feeling where is what the genotypes, how the growth, how the condition is with respect to growing, height, biomass, everything. You can see here, there is a dip here, there is a height variations here, there is a fallow here. So everything you can capture and try to retrieve them, and you can see the heat map generated over using different indices. So instead of going to a model field to capture the data, which takes a dates together. So if you tell you have a technology which were expertise to retrieve the information using a regressive model for the different parameters, you can go for the heat map generation out of this. So you can think of the kind of 10,000 land races. If you tell you want to go for a chlorophyll measurement or a height measurement, how much time really it goes for. So this is what the kind of opportunity with respect to technology, advancement, and availability, what always one can use for. So coming back to next one, sorry. So this is what, again, the machine learning or deep learning programs where you can, this is what in the phenomics image captured, RGB image you captured, and you are able to see the number of barnacles, how it is detected using a deep learning program. So like you use your RGB camera while doing photograph, you get the boxes in your mobile, and that box focuses your sensor to the detectors, the person's face. Same also here, the same technique we try to use. So number of barnacles you can always predict go for. So same thing we try to do with the drone. This is a Institute of Mage Research. They have a field experiment in the IRE field itself. We try to capture the data with RGB and try to count all those tassels available in that field and number of tassels, how they're different with respect to different response that can be done. But number of tassels you could count here in the field conditions flying a drone, so which otherwise is a time taking. And so we try to see also, so there are some other experiments. What we have is recently a precision agriculture program where you try to evaluate different sensors like RGB. You have a multispectral, you have a hyperspectral, how they're good or bad. So you look at the RGB, this is what the 3 bands you have. When you look at the multispectral, you have 5 bands like this. When you look at the hyperspectral, this is what the hyperspectral, the green one, 272 channels. And when you have a sensors like up to 2500, for example, this is a sensor, of course, non-imaging, this is around 2151 bands. So think of having simply RGB, having 3 bands, you are deriving so many informations from the field itself. You have been doing many research using simply RGB camera. So when you think of multispectral, you are deriving so many. So when you have a hyperspectral, you can derive the information which is beyond imaginations. I mean to say all kind of traits you can capture too. So for example, look at the hyperspectral data, what we try to do. So this is what the sensor capture the data. We try to stitch them together and generate the data. And this is what the spectra you are able to receive. And these are the number of indices you always can calculate, around 61 hyperspectral indices, which was calculated using the software, as I told you. So then you can put them together, come out with what is that sensitive index, particularly for the nitrogen predictions that we try to do. I'm not going to talk all in detail. This is what try to do the feature selections to find out how the prediction of nitrogen can be done. This is what the prediction of nitrogen is done. This is what the nitrogen treatment given in the field. This is what the leaf nitrogen concentration, which is done by wet chemistry. And that's what the matching also we try to do it. So you have a regressive model, as I told at the beginning. Now you have some of those machine learning programs we try to use for this purpose using different indices. You can see here, these are all machine learning models. For example, artificial neural network, extreme learning machines, support vector machine, lasso, and of course, MLR, and try to model that and come out with the predicted nitrogen. So if you at all leaf nitrogen is your attribute or chlorophyll content is your attribute, you can capture and see the differential response also in the field conditions. Same also for LI, we try to do with the machine learning programs. And you can see, so this is what the different predictor model for the leaf area index, leaf chlorophyll content, and also canopy chlorophyll content, which we could capture and see the variable response from the field with the differential treatment. Of course, these fields were treated differently with respect to irrigation with the nitrogen to see how we can create the variations, and that variation is captured by the sensor, and how that models can be used for this purpose with a good accuracy. Some of those models you could see here. I think our conclusion was these are all published work right now in the photogrammetry and remote sensing journal. So you can see mostly artificial neural network is really overdoing with other techniques. So something around 0.99 is coming. So this is what the kind of capability what has been developed with time. So again, this is a hyperspectral sensor which was used in the drone, and you are seeing the differential treatment here with respect to the nitrogen. You are able to see that color map with respect to nitrogen concentration here. So this is what the spatial variable map you could capture, and each field is altogether treated differently. For example, this field is with the N0, you are able to see that variations. Again, also within the field, that kind of variations you are able to see. So these are the techniques which can be used for retrieving different attributes, what you are looking for. This is again a little advance of the technique. For example, you look in different directions of a plant, and plant has a geometry, plant has a morphometry parameter, like you do photography of yours. And when you do photography in different directions, the photography is going to be altogether different. So this is what the new science try to do. Also, we have a couple of students who have already done this work. We try to use a goniometer to do that kind of a capture. And you can see the same soil, the signature is the radiation is getting different. Same crop, radiation is getting different in all possible directions with respect to sun. So what it fails here is, whatever the assumptions we have till now is that when you take from the nadir, you capture the image, everything is done. No, it has a directional property. Each object has a directional property. In a particular directions, you have more radiations or less radiations, and you can capture more information or less information. If you total, you maintain that directionality. So that's what we call it BRDF, bidirectional reflectance. And that's what the kind of research we try to do. And same also we try to do with the drone, to look at what directions, instead of flying nadir, If in a particular directions I can capture the data, then a lot of morphometric parameters I can capture. For example, look at the wheat varieties of different height, different phyllotaxy, different Li distributions. That can be captured wonderfully, and differentiation can be done wonderfully if at all you use this kind of technique. That's what the purpose we are doing too. Then if at all use the same technique, like till now what I was talking is regressive model, machine learning model, this is a process-based model. Process-based model tells you how the energy comes in, interacts with the soil or plant, goes back, and understanding the physics or chemometry, how the radiations interact with the different biochemical parameters. You can simulate the spectra. Also, you can retrieve the parameter what you're looking at. So this is what it tells you. You can simulate the spectra sitting in the lab itself. How, if at all I go to a field, having a sensor, how my sensor is going to behave. This is what simulation you can be doing, understanding the physics and biochemistry behind it. So this is what called radiative transfer modeling. And if at all I have a data here, like from a hyperspectral or from multispectral, from satellite or drone, then I can retrieve all these parameters by inversing. So this is what the new approach try to come out with. If once you fly the drone, within no time, you should be able to retrieve the parameter you are looking at, maybe Li, chlorophyll, or water, whatever you are looking at. For example, so I think I can skip all these slides. So this is what it tells you. This is what the simulated spectra, what you have generated using the similar model. So a lot of physics goes into that, and also understanding the interaction of the radiations or energy with respect to different parameter, chlorophyll, Li, or the water. And this is what the simulations you are able to. And once you do that, then you can revert the model and can generate that parameter like Li, chlorophyll, or water, whatever you're looking at. This is what it is retrieved. Why I'm saying this, once you do this Li prediction is going to be more accurate, more generic. It doesn't need any coefficient to change the model. So this is what the kind of science can be done with. Now, coming a little bit with respect to that is with respect to retrieval of the traits or attributes. Now coming back to the reals, the genotype discrimination or capturing them with respect to the parent. For example, you have so many 278 reals, which is a parent is RAS 3765, SG 2329. You know the best attribute what they have. And we try to evaluate these 270 reals with respect to their parent, which is matching. Very interesting results we could get over here is you can see the different stages data what has been captured. And as I told you, this whole spectrum is a function of all the attributes you are looking at. Attribute is a leaf area index, chlorophyll, height, biomass, or whatever. Now, what you do is conventionally you measure them and go for the matching with the parent and see how best they are matching. Then you look for the QTL for those purposes. But here, I want to go for simply matching of the signature of the parents like RAS 36765, SD 2329, and also with the reals. Once it matches, I think that tells me I don't need to go for chlorophyll measurement or nitrogen measurement. Then I can go for the matching and similarity. Once the spectral matching is done, then I can say this reel is really contributing to same as what the parent one or parent two or both the parents' best attribute it has taken. So this is what the exercise which has been done in the field conditions. We are yet to publish this work. So this is what the similarity capturing is done. Like with respect to parent, for example, this is P1 is the RAS, P2. So this is what the matching has been done with respect to something around 278 genotypes, which really is matching to both the attributes that can be mapped. And you can see some of those variations with respect to the matching similarity. So this is what the kind of science what you can do. You can go with weight chemistry once you understood how well related the weight chemistry data with the signatures. So this is again a data which you can visualize and interpret with the time, how best they're working with. I think let me play with. So this is a field having the different treatment of the, like this is a field with the water stress. This is a field with the recommended doses. This is a field with the nitrogen stress. And this is a different dates of data capture is given down here. And you are able to see you threshold, take out the soil part, only the crop part you can keep. And you can see the kind of matching how it does. So this is again different date, how the behavior is. You can see the response. You can see the differential response with respect to the visualization, how the field is behaving, and also the field level variations and also genotype level variations. Again, you have a different phenology, different data capture, which you can see. you can see wonderfully how they are behaving. The central one is the recommended doses of nitrogen and water. Left and right is treated with the water deficit and nitrogen. So this is what the kind of data analysis, visualizations, what can be done within no time. Once you come back from the field, you can do that kind of using multispectral, also even if thermal. Coming back to the discrimination, I think I have a few minutes more. So this is what the rice varieties with the basmati, non-basmati, also different aromatic, non-aromatic rice. I remember long back, Dr. G.P. Singh and myself and Dr. Prabhu, we had an experiment. He had a 70 wheat genotypes, and that was the first experiment we tried to see if at all we can discriminate those 70 genotypes using the hyperspectral remote sensing. I think that was the first work we did in the discrimination, because then the then director, Dr. H.S. Gupta, was asking whether we can map what are the IRE varieties grown in the field conditions, what is the mapping area. Like same also, what is the area where the basmati growing regions, and how much is the basmati growing regions with respect to particularly varieties developed by IRI. So that was the question always is there, how we can address that. So to keep in that, so we did also some kind of studies, as I said, with the Dr. G.P. Singh, who is your very dynamic director with MPPGA, and we have been working with him when he was at Canal also. So you can see the kind of discriminations, genotypes, what we could do using again same spectral separability analysis, and also we try to do same technique we try to use here with respect to hyperspectral data. So this is what the field which was grown with the many genotypes, and we try to capture this data, and you can see this is what the field with the different genotypes with respect to basmati, non-basmati, and basmati. You can see the color that tells you which is basmati, non-basmati. So you could see the differential response with respect to basmati and non-basmati, and that is mapped. Again, you can see aromatic, non-aromatic. These are the classes. Which, this color tells you the aromatic, non-aromatic. And you can see the discrimination again. You can see the spectral changes, how it is. So again, look at the genotypes of basmati, non-basmati. Again, their differentiation, almost all those 29 has been discriminated. You can see the spectral behavior altogether, they're different. And so this is what the interesting, and again, near isogenic lines, also you could see their difference with respect to those basmati, non-basmati areas. So what I mean to say is the technology is there to see how we can get that differential response with respect to crop growth, which visibly is not available, but you can see the spectral response, and you can spectrally differentiate them like your signature. Each individual, your signature is altogether different. So your fingerprint is altogether different. So that's what the biometric we do. Same also, this is kind of a biometric of the plant, and that can make you differentiation. These are some studies related to diseases, pests. So we try to do in the lab conditions. We try to do it also in the field condition. This is a rice blast. That was a BPH. This is a yellow rust. Again, we are done with in collaboration with the Canal. And this is the field, again, Cuttack, where we have flown the drone and also gone for the variable spraying. I think this is beyond the. So this is what the drone flown here. Down one is the hyperspectral camera going for the nitrogen mapping, the pesticide and applications. And the top one is the spraying where it does. You are not able to see them. The mist they are in the same color. So this is what the spraying is done. So then, so why only drone remote sensing? There are, you look at the Washington DC University, you look at Iowa State University, also Purdue, the guys who have been working very high resolution satellite image for field phenotyping. Same also we are trying to look at. This is what the pipeline methodology we try to develop in the IRE field mapping. I think I should skip all those things. Let me come back. So this is what here, the same pipeline can be used. For example, you have a drone image captured with the multispectral and hyperspectral, and you can see what the differential response in the field conditions. And you can do in the drone conditions, and also you can do from the satellite, provided you have a high resolution satellite data. So again, I think I should, this is what the concluding sides of what I mean to say here is, there are opportunities with respect to the sensors and platforms, but there are opportunities also using this machine learning and deep learning programs, how best to design different genotypes, design their differential response with respect to Niels or reals or genotype. There's a really challenge. Genotypic level, plant type level of discrimination is much easy. When you come back to the genotype level, there are challenges. They're not necessarily always it will be matching. Again, when you come back to the reals, next to difficult. That's what we could feel. But somehow there is a positive hope we are going to come out with a technique or technologies to see how differential response we can come back with respect to real-time inferencing and using high data analytic tools and modeling, and of course using this high resolution data for the field phenotyping. And thank you very much. I think this is a team in IRA with a multidiscipline from the genetics, from the plant physiology with Dr. Vishwanathan, Dr. Gopal Krishan, many, also NBPJ, Vikender is there. We have a experiment in NBPJ field. We have been capturing a lot of data, and also Kuldeep Tripathi and also Kamal Mahapatra. There are people who we have been working together. That's how I'm able to show some of our experience, what we have done with. And thank you for patience hearing. I love to interact and take up some questions if at all. Thank you.
Thank you, sir. Thank you. Dear participants, if you have any questions or queries, please ask to speaker. Otherwise, you can share in chat box also.
There is one question in chat box.
Yes.
Cost is constant to replicate ubiquitous phenomical facility. Sir, there is one question.
I could see that. Let me
Yes.
put this is ma'am, Dr. Subha Mongali.
Dr. Suma Mogali, sir. She
Yes, asked for cost is a constant to replicate ambiguous phenomic facility. That's perfectly fine. So you don't need to go for replicating Nanaji Deshmukh Plant Phenomics Center. You spend 20 lakhs, you can do high throughput field phenotyping, and that's what the end of the that's what the requirement of the day. So you need to just spend little money for field phenotyping, which otherwise you spend crores of rupees in the wet chemistry and also field data acquisition with respect to labor and kind of energy you put in. So I think it's a very cost effective if at all you go for field phenotyping using a mobile platform like we have, or a drone. So you don't need to look for the Nanaji Deshmukh Plant Phenomics Center, which is a 47 crores facility. You don't need to look for that. That's a basic question to be answered. But in field phenomics, of course, you don't need to invest that much money.
Sir,
Okay, share the presentations. I'll share with Dr. Ravi. I think it
Yes,
will be
sir.
available. I think this is recorded.
Yes, sir. It is YouTube also. We are
Yeah, perfectly
posting
fine.
on
I
YouTube.
think
Yes.
that meets the requirement. I think.
Sir,
I think no question.
Another question from Dr. Suma.
what is the regression analysis experience, sir?
Experience, sir, in a comparison of visual phenotyping.
Yes.
Oh, visual phenotyping, I could show you the way it is. Like, for example, we did in case of wheat, we did in case of NBPJ experiments also, like how much germination has been taken place or how much flowering has been done. That is a visual, anything visual is a qualitative analysis. You won't get a exact figure. But when you go for the technique like regressive or the kind of a computer vision to count number of flowers in the plant itself, a number of tassels, that is going to be more accurate and better. And to always a visual is a subjective matter. So that's what the basic difference. But to do a quick analysis, always you can visualize and make it appeal to see how wonderfully the genotypic response with respect to their germination or flowering or attending a particular stage is done. When you capture the data, you can do that. So because of the time constraint, I didn't talk much about how to do that. And there are the census has that kind of a capability to capture even if phenology stage also. Like what stage is the phenology has come particular genotype that can be captured using some of those techniques. So always it is a quantitative analysis is always much better than visual or qualitative analysis. I think you got my point.
Yes. And any other participants? Sir, I hope there is no other doubts. And dear trainees, if you have any doubts, you can chat in post box. Chat in our chat box. or otherwise you can send request email to us. We can share with sir and sir will give the answers.
I have given my email ID there itself. I'm just right now ID.
Yes, sir.
So that anybody can do that.
Yes, sir. Sure, sir. Sure, sure. Sir, I will post your email, sir.
I am posted.
Yes.
The chat box itself.
Yes.
We'll
sir.
also
Thank you
share,
very much,
sir.
sir.
We'll also-
नमस्कार सर.
Oh, namaste. कैसे हैं आप?
बढ़िया सर, बढ़िया सब ठीक है सर. बहुत अच्छा लगा सर सुन के, जो काफी कॉम्प्रिहेंसिव लेक्चर सारा कुछ कवर किया सर.
Yes, sir.
Thank you.
सर व्हीट का एक्सपेरिमेंट सर ईसापुर में भी लगा हुआ है, बहुत बड़ा सा. न्यू एरिया में तो छोटा है, तो ईसापुर में भेज सकते हैं सर.
ईसापुर में मैं डॉक्टर कमल महापात्र के साथ क्रॉप द you have a trees, no?
Yes,
There
sir.
are,
Three gene banks, sir.
biodiversity mapping we have done. We are doing on that also,
Okay.
with the RGB,
sir.
multispectral, hyperspectral. We visited once with the courtesy of Mahapatra and also
Okay.
ICRA. We can do, provided we have a time for that.
Yeah, wheat ka means we have large experiment there. DVT trial jo we are conducting there.
We can see.
A small
We can-
experiment is there.
We can see, no issue. I think there was a proposal. I think I don't know Dr. GP Singh is here or not. When Dr. Kuldeep was here, there was a proposal for Center of Excellence on high throughput field phenotyping, and drone remote sensing was one key factor. And we proposed that, me and Vishwanathan. If at all we create a Center of Excellence for that purpose, you guys know best to how to do the field level, all those things, and we will use our expertise to combine together, come out. That proposal has gone to the ministry, to the DBT, but I don't know what happened.
Okay.
Namaskar, sir. Malik sir.
Sir, welcome.
नमस्कार सर, मलिक सर नमस्कार.
नमस्कार डॉक्टर साहब.
Namaskar, sir.
How are you?
Good afternoon,
I'm fine.
sir.
Nice to see you after a long time, sir.
Yes, sir. Okay. Okay, ma'am. Sir, it is a time to say vote of thanks. And I really thankful to you for briefly explain the drone-based technology in agriculture. And especially, sir, it is a wonderful presentation in reels of wheat and basmati rice, as you take the case studies and explain the how we can use the drone-based technology in particularly in assessment of nutrients and other water management. And sir, really, sir, thankful to you. And you given some challenges also, and you explained in our concluding session, sir. That is especially that is an unmanned aerial vehicle technology protocols. Also, you mentioned, sir. That is really wonderful, sir. Sir, really thankful to you, sir, on behalf of organizing team and other members, the participants. Thank you, sir. Thank you very much. Thank you, sir.
Thank you. Thank you, sir.
Thank you, sir.
Thank you. Sir.
Thank you. Thank you for giving me this opportunity. Thank you.
Thank you. Sir.
Bye-bye.
And dear trainees, we have next speaker, Dr. S.K. Malik, sir. And Dr. Subhash, please, over to Subhash. Yes.
Thank you, Dr. Ravi. Welcome, sir. And good afternoon, sir.
Good afternoon.
How are you, sir?
Good afternoon. I am
Good afternoon,
fine.
sir.
Good afternoon to all of you.
Yes, sir. Good afternoon.
I am
So,
audible?
sir, there is a
Dr. Subhash,
time
I'm
is
audible?
12:30 or we can start.
12:30.
Yeah, time is 12:30. We can start, sir, because
Okay.
all are present.
Okay.
You can continue, Dr. Subhash.
Yeah, we can continue.
Okay. Okay.
Okay, sir. Let me share this.
Yes. Okay, continue.
Sir, good afternoon and good afternoon to everyone. Sir, I formally welcome you, sir, in our to join with us. Friends, we have another lecture by Dr. S.K. Malik, sir, on conservation of non-orthodox seeded germ plasms using cryopreservation technique. Let me introduce our speaker, Dr. S.K. Malik, sir. He is a principal scientist in Tissue Culture and Cryopreservation Unit, ICR NVPG, New Delhi. Sir has vast experience of more than 25 years. He is working on management of PGR, reproductive biology, organogenesis, morphological and molecular characterization, in situ on-farm conservation of horticulture crops, including fruits, plantations, and vegetable crops. Sir has conducted more than 60 exploration and collection, and from northeastern India, Western Ghats, and other parts of India, and more than 7,000 accessions of non-orthodox seeded species, and more than 400 accessions of dormant birds and pollens were conserved in cryogene bank. Sir has established a comprehensive cryo-based collection of Indian citrus, germplasm, underutilized fruits, plantation crops in cryogene bank. Sir has also developed a cryo protocol for citrus and non-orthodox seeded species. And first time, sir reported the chromosome data of citrus asamensis and citrus aikigensis. A new wild type of citron, that is Citrus medica, were identified from northeastern India through morphological and molecular markers. Sir has proposed an in situ on-farm conservation model for wild species of citrus in northeastern India and underutilized fruits in northwest India. Sir has established 22 community seed bank in district Udaipur and undertaken in situ on-farm conservation efforts. Sir is actively involved in MSc and PhD teaching of especially PGR discipline in as a IR faculty, and he has more than 20 years of teaching experience, and he has guided 7 MSc and 5 PhD students. Sir has published more than 200 articles, including research paper in reputed journals, and sir has authored books and written book chapters, popular article and technical bulletin and brochures. Sir has conferred with the fellow of Indian Academy of Horticulture Sciences and Indian Society of Plant Genetic Resources. He is editor of Journal of Biochemistry and Biotechnology, published by Springer, and sir has conducted international training. More than 7 international training has conducted on in vitro conservation and cryopreservation of PGR. That is under ICR Biodiversity International Center of Excellence. And sir has overseas exposure, and he has visited IPK Germany in 2015 for training and scientific exposure under bilateral cooperation between India and Germany. Sir has also visited universities of European nations, including Netherlands, Germany, and Switzerland, under the Executive Development Program on developing effective organizational leadership for senior officers of ICR. And sir has a lot of experience in cryopreservation. Sir, please share your expertise and your vast knowledge with our participants. Now to you, sir. Please share your slides.
Thank you, Subhash. Thank you very much. Is it shared?
Sir,
Not yet, sir.
please,
Not yet.
sir, first you open your slides, your presentation, then you share, sir.
No, it is open.
But sir, it's your desktop is shared, sir.
Only desktop is shared.
Yes, sir. सर पहले अनशेयर कीजिए, फिर उसके बाद में आप अपना पीपीटी ओपन करके फिर दोबारा शेयर कीजिए, पीपीटी को शेयर कीजिएगा.
Share screen. Is it okay now?
Yes, sir. Now it's okay, sir.
Okay, sir. Okay.
Okay. Thank you. Thank you, Dr. Subhash.
Welcome, sir.
So, my namaskar to all trainees and other participants of this training. First of all, I thank the organizers for giving me the opportunity to present this lecture and share my experiences with all of you. So as you can see, the topic of my talk is cryopreservation of non-orthodox seed species, practical consideration. So I will be more emphasizing on the application part of cryopreservation, including cryobanking. So this is the outline of my talk. I will little bit talk about the PGR conservation, cryopreservation, seed storage behavior, then collection and handling of non-orthodox seeds, application of cryopreservation techniques, and finally I will be showing you a few slides of cryobanking, how we do the cryobanking of germplasm at NBPGR. And I know that you already had a few lectures on cryopreservation during your first week of, I think, this training. And maybe few things you will feel as repeated, but once you are at the final stage of the training, you just treat them as just a repetition or just as a reminder, or these will just remind you what you have earlier learned. चेंज नहीं हो रहा है. Okay. So PGR conservation strategies, already you know that how we are doing it. This is the in situ, ex situ. There are several methods in situ. And when we come to ex situ conservation, in laboratory, we see the seed gene bank, cryo gene bank, in vitro repository, and genomic repositories. So in this particular lecture, major emphasis will be on the cryo gene bank. So I will be emphasizing on the cryo gene bank, how we are doing especially the non-orthodox seeds. So when you talk of the non-orthodox seeds, basically these type of germplasm belongs to the horticulture species. And what are our different candidates which we are actually targeting when we talk of the non-orthodox seeds? These are the several cultivars, clones, varieties of horticulture crops, including vegetables and the fruits and all. Then we have the rootstocks. We have semi-domesticated types. Some of the species, you will be seeing some of the examples where we have applied cryopreservation techniques to the semi-domesticated species. Then we have the natural wild. So in these all four type of the candidates, what we are preferring to preserve is these are the different explants which we are taking. Here we can take the clonally propagated species, then preferred explant is vegetative tissue. And in rootstock, generally we do the preservation through clonally as well as seed, through seed. And in natural wild, mostly we try to preserve as a gene pool conservation through seeds and embryonic axis, and similar way in the semi-domesticated types. So just coming to the cryopreservation, already you know the definition. It is a non-lethal storage of tissues, especially the biological material, at ultra low temperature. And when we talk of the ultra low temperature, it is minus 150 to minus 196 degrees Celsius, generally in the temperature of liquid nitrogen, or in the vapor phase or liquid. Both we use for the biological tissues. And the major explant which we take is the seeds, embryonic axis, buds, and cells in case of plant germplasm. And there are several methods available nowadays, and these are the, especially we follow the two freezing techniques, that is slow freezing as well as in fast freezing. And I will be discussing a little bit about the fast freezing techniques, what we apply to the generally seeds, embryos, and embryonic axis. Just to show you the why actually it is so typical when we talk of the non-orthodox seeds. Because in orthodox seed, you might have seen that we can easily preserve the germplasm at minus 20 degrees Celsius. We can desiccate them, the seeds, and then we can go for the minus 20 degree Celsius preservation in the traditional gene banks, following the standard protocols and all. But when we talk of cryopreservation, actually what is happening is that when we remove the water, and we need to ensure that no crystallization is taking place. And these are the different steps when we follow, and we need to see that cells and tissue survives. So for that, all different techniques are used, and especially the slow freezing and ultra rapid freezing to avoid the crystallization when as far as the freezing is concerned. So desiccation and freezing, these are the two major parts when we talk of the successful cryopreservation. So development of cryopreservation protocols, we need to know the morphology and the physiology of the explants. So these both are important. As I discussed earlier, the desiccation and freezing, tolerance of the tissue of the cells is important. And when we are moving to the cryopreservation, we should know the morphology and structure of the explant. This is must. Then sensitivity to desiccation. This is to be standardized during the protocol development. Then sensitivity to low temperature, how much low temperature we can put the material. And then longevity of the explant. This is again very important. When you see that in nature, there are several types of the seeds and tissues which have the different longevity. I will show you some of the examples. And to standardize all these, we should have the optimal technique, which can be used to cater all these parameters. So when we talk of cryopreservation of especially the horticulture crops, we have the different explant like seeds, embryos, embryonic axis, dormant buds, shoot apices, pollen, and somatic embryos. So generally all these explants are being used. And especially in this particular lecture, we will be going to discuss the seeds, embryo, and embryonic axis. Now coming to the seed storage behavior, here you can see the first time in Roberts 1973, he gave the concept of seed storage behavior, and he enumerated that these are the two type of behaviors like orthodox and recalcitrant. And orthodox seed is desiccation tolerant, and recalcitrant is desiccation sensitive. Then again in 1984, Barjack given the different name because she and her group was working on especially on the mangroves in South Africa. And they actually called the orthodox seed as a poikilohydrous and recalcitrant seed as a homeohydrous. This was just the terminology was changed, but the characteristic of the seeds was the same, that recalcitrant and orthodox. And there was a one turning point in the 1990 when Alice et al. Given the different one new category of the seeds, which is intermediate types, like in citrus and coffee. They said that there are some seed behavior, some of the seed which behave in between the orthodox and recalcitrant. But now I can tell you, after the long experience working on the different type of seeds of tropical nature and temperate and others, so there is a degree of recalcitrant. There are several papers published now of Pritchard and so many other groups. And there is a orthodox, which are the desiccation tolerant. In between, when we say the intermediate, and highly sensitive to desiccation and freezing are the recalcitrant. So all this is due to the presence of high moisture content in this. When we see the degree of recalcitrance, it will vary in the different seed types. You cannot say that one absolute, in absolute terms, that this is orthodox, this is intermediate, and this is recalcitrant. So degree of recalcitrance varies. When the seed is immature, it will be having the high moisture. And when it have the high moisture, it will behave just like the recalcitrant seed. Even the orthodox seed, when it is immature, it will behave like the recalcitrant or intermediate seeds. Like one of the species here, I have mentioned is Citrus ichengensis. In most of the seeds in citrus, the genus citrus, most of the species have the non-orthodox seeds or intermediate type of seed. These are not exactly orthodox or highly recalcitrant. But one species, Citrus ichengensis, it is having the very large seed and it behaves as a recalcitrant seed. And another aspect is the vivipary. Here you can see the seed already started germinating in the fruit. So this is the exact characteristic of the highly recalcitrant seeds, which there is a very less time between the seed drying as well as the seed starting to the next generation, to the formation of the seedling and all, again germination. So here, when the seed is fully mature, it get the moisture content inside the fruit and it start germinating within the fruit. So this vivipary is very common in most of the recalcitrant species, like you might have seen in jackfruit also. Vivipary there, and several other species, like especially in the mangroves, like Avicennias and other. This is very common feature. So handling such a type of seeds which don't have the desiccation mechanism, intrinsic desiccation mechanism in them, just like the orthodox seeds. You might have seen the seed of several cereals, millets, and pulses, which when the seed dries, and you can easily store that seed, and during the next generation you can sow, and when the water is available, those seeds will germinate. But this is not the case in the recalcitrant species. So these become really very difficult to handle, these seeds, for the conservation. Just to show you a few examples, like in the recalcitrant seed itself, you have the several categories. Like in Garcinias, most of the seed are apomictic. There are the both type of, some of the seeds are facultative and there are obligate agamospermous. And I will not go into the detail of the reproductive biology of these. But here you can see the seed size is very large, and even handling of the small pieces of seed due to the high moisture content is difficult. This is the special type of behavior in the Garcinia, where each part of the seed, each piece of the seed can make the plants due to the heavy, these highly totipotent tissues on the surface. So these are the seeds which we handle. When we handled the first time, we were really shocked to see that one part of the seed is giving the root and another part is giving the shoot. So there is a polarity in the seed. So this is a special character in Garcinia and some of the other members of Clusiaceae or Guttiferi. Then in polyembryonic seeds, like in citrus, most of the species are polyembryonic except 4 species. So you find these type of difficulties that nucellar seedlings are also there. Like in jamun also this phenomena is there, and several other tropical tree species you will find the polyembryony. Then high volume seeds, as I told you, this is the citrus icen, this is fruit. Is full of seeds only, very large seeds you can see here, like in Pogemia and Maduka, Mahua, jackfruit, the big, large, very large seed here you can see. And here again you can see that the seed had started germinating within the fruit. This is the complete fruit of the Mahua. And in some of the species, you will find the unexpected seed storage behavior and longevity. And one of the major example which we studied for last several years is the Salvadora. In the local in Haryana, Punjab, and some of the drier part of the world, this is there are the different species are occurring. This is Salvadora oleoides and Salvadora persica, especially which these occur in Indian desert and especially in the western part of the country. Here, this we found that this is a seed which is, this is the fruit which is occurring at the minus even the 50 degrees Celsius temperature in the Thar desert, and seed is maturing after drying or at maturity. This is completely dried. There is not much moisture content, but the seed just behaves just like highly recalcitrant. So there are some factors within the cotyledons, in the seed cotyledons, which are making this seed highly recalcitrant and very short-lived. So this is the TTC test you can see, and how the seed is behaving. Within a few hours and few days, it is totally dying. So we have excised the embryonic axis and did some experiments and could successfully cryostore it. Just to show you one example of Garcinia, you can here just see the this is the fruit, this is the seed with the coat and all. This is with uncoated, and this is the part of the seed. Both the ends actually we have taken, because both the ends have the meristematic tissue, and these are the active parts for cryopreservation. So we can use the some small explants, excise like embryonic axis, and do for go for the cryobanking. These are some of the examples of the intermediate seed storage behavior, where you can desiccate them 10 to 12% moisture content, and then go for the fast freezing to get the success. I will show you some of the examples where we have successfully cryopreserved the several species of underutilized fruits and citrus. These are some of the examples where we found the intermediate type of seed storage behavior, like in papaya, tea, almond, neem, star fruit, Arabica coffee, several species of citrus and oil palm and others. So coming to the how you will see the seed storage behavior, this is the actually protocol which was given in 1996 by Hong and Ellis. And I will not go into the detail of it, but I will just explain here that moisture content and initial viability are the two criteria which we take. And to ascertain the seed storage behavior, we can reduce the moisture content to 10 to 12% and then go for the testing of the viability. If there is no survival at this particular moisture content, these are the probably recalcitrant. And if there is maximum survival, we can desiccate to 5 or 6 or 7% moisture content and then test the viability. If there is no survival, we can just treat it as a probable intermediate. And if there is a survival and the seed can be stored at minus 20 degrees Celsius, we again test the viability and treat those particular type of seed as the orthodox seed. So using this simple protocol, we can decide that seed is orthodox, intermediate, or recalcitrant. And then we have to control the moisture content and initial viability we have to see. And generally in the recalcitrant seeds, the moisture content is generally above 30% of and more. In generally, some of the cases we found that it is 60 or 70% also. So this is the major problem, how to handle these seeds. So I have made some of the steps. So to how we can actually do the proper conservation. And a very important aspect is the post-collection handling. Collection itself is a very difficult. Collection and the post-collection are the two typical aspects to successfully conserve these non-orthodox seeds or recalcitrant seeds. So post-collection care and field and laboratory handling of recalcitrant intermediate seed is difficult. As I told you, the characteristics of these recalcitrant seed, that they are very shedding at the very high moisture contents. Seed storage in a large number of cases is unknown. This is again the phenomena. In several of the tropical and temperate species, still there is a seed storage behavior is not properly studied. And longevity and storability of the seed is also unknown. And precautious germination occurs in several cases, as I told, in jackfruit, cocoa, citrus, and several other species. The large seed size and limited availability of seeds, because especially in the tree species, you will not get the sufficient number of fruits to extract the seeds. If you get the fruits, the number of seed in the fruit is limited. So you won't unable to get this sufficient material for your experimentation even. Then difficulty in getting desirable accession size to conserve total spectrum of genetic variability. This is again becomes very, very difficult. If you want to conserve the whole spectrum of genetic variability, you need the large sample, which is not possible here. Then availability of protocols, in vitro conservation and cryopreservation. In some of the cases, you have to regenerate or you have to do the experimentation using in vitro techniques. And in most of the cases in recalcitrant seed, which belong to the tropical tree species, it is difficult to establish in vitro, and growth is very slow. And there are several issues which I will try to show you some of these. So improper collection and handling of material results in low initial viability and seed health due to the very high moisture content. The factors contributing to these are poor understanding about the morphology and physiology of fruits and seed, collection of fruit and seed at physiological maturity, not suitable for conservation. So in some of the studies which we have found that if the seed is collected at the right maturity stage, you can successfully cryopreserve it. If seed is highly matured or it is immature, so developing protocol and cryopreserving becomes difficult. So this is we have to decide during the protocol development that what stage, physiological maturity stage, is good for the developing this material for the cryopreservation. So inappropriate seed extraction and storage. This is again one of the very important things. As I told in earlier slide, that morphology of the explant is very important to study, because these when you talk of the especially the embryonic axis, these are deep seated in some of the species. Excising the embryonic axis, it itself becomes very difficult. Even in some of the species, the extracting seed from the fruit, it's become difficult. And its storage then again is a very tricky. And use of inappropriate method for seed drying, because as I told that we have to reduce the moisture content for going for the freezing. Before the freezing, we have to reduce the moisture content up to the optimal level. And in each species, there is a different type of moisture level which we have to standardize. Then the infection of material by saprophytic fungi and bacteria, these will, due to the high moisture, these things will happen generally. And conservation of pest and disease-free germplasm is difficult. So how to go about, how to address all these issues which we are facing in this? Briefly, I will just explain. And fruits and seed may be collected from tree marked at the time of flowering. We need to do the two exploration at 2 times. First, we need to mark the trees which are healthy and give you the sufficient number of fruits and all. Then harvest the fruit directly from the tree. Then maturity of fruits may be divided into 3 developmental stages. That is immature, partially mature, and fully mature. Generally, in some of the fruits, we have done these experiments and found that sometime the partially mature seeds are more amenable to the desiccation and cryopreservation. So these type of things can be attempted. This is in the case of cocoa seeds. Here you can say this is highly immature, but embryonic axis of the seed is very healthy here. Then in partially mature, this is again cocoa. You can just see the growth of the embryonic axis which has taken place, and this is the fully mature. And in we have actually had one externally funded project from UK, and on cocoa, jackfruit, and lychee, and a lot of experimentation we have done in that, and we could find that partially mature stage is more suitable for the desiccation and preservation. And seed development in desiccation sensitive. Again, this is what I was emphasizing again. And lack of ability to reverse when rehydrated. When you are drying the seed, in some of the seeds or tissues or cells, there is a very limited ability to reverse that trend. While in orthodox seed, you can reduce the moisture content, and when you give the moisture to those seeds, those cells will revive. So that phenomena, that physiological understanding, that physiological characteristic is not there in these seed. So these are some of the examples of the underutilized host which we have actually undertaken, like Carisas, Phoenix, Manilcara, Exandra. These we are collecting as a fruits directly from the trees, and then we are bringing it to lab and then handling. Then fresh food should be transported immediately after collection to the laboratory in cloth bags. In case of it is difficult to transport freshly extracted seed packed in sawdust, we can pack the seeds in the sawdust and charcoal or peat moss, and then we can transfer them within 48 hours. Then record fruit color, firmness, and other data we need to record when we are handling some of the new species. This is we can do it like in fruits. If it is possible, we can collect the fruit and bring, or we can extract the seeds, and these seeds can be brought to the laboratory. In laboratory, fruit seeds may be stored at 15 to 20 degrees Celsius, not at the lower, very low temperature or very high temperature. So this is the ideal temperature where we can keep our fruits and seeds during the experimentations. In case of whole seed is not being processed for cryopreservation, we have to excise the embryos and embryonic axis. In most of the cases where the seeds are large, you have already seen in some of the species. So we try to handle the embryo and embryonic axis. And embryos and embryonic axis being deep-seated generally do not carry any infection. Hence 8 to 10 minute treatment with a sterilant is sufficient. So we have to sterilize the seeds, and then we go for the excising the embryonic axis or embryo out of the seed. The most convenient and quick method for excising embryonic axis need to be devised. I will show you how we can do it. And in the different species, there is a different technique which you need to apply. And for extracting the embryonic axis, you have to be very efficient. So maximum number of embryonic axis you can excise in the limited period. Then moisture content of seed, embryo, and embryonic axis should be measured using low constant temperature oven method. This is actually what is suggested by the ISTA. So just to explain you this particular part, then in case of whole seed like this, if we are handling the whole seed, there is no issue. We can directly go for the desiccation and freezing and all. And if this is the embryo, this is without seed coat. This is in some of species of citrus, and this is then embryonic axis. And each seed, you will find the location of embryonic axis would be different. So that you have to first study the morphology, see where the embryonic axis is located, and then devise the proper methodology how to extract it. So this strategy or pre cryopreservation methods need to be devised. And these are some of the factors of successful cryopreservation, that cytoplasmic non-vacuolate cells or embryogenic and meristematic cell most amenable. And this is why we prefer the embryonic axis or embryos. And you see the whole seed, there is a lot of other tissues are available. When you handle the embryonic axis, you will find the most of the tissues are meristematic, and these are having the very small cells full of cytoplasm and active cells. So these are more amicable to the cryopreservation or desiccation as well as cryopreservation. And dehydration level of the each explant that we have to see that at which level we can get the maximum viability. So that particular moisture level, we have to go for the next step of desiccation. Then cooling rate, as I told you, fast and slow freezing is also plays very important role. Then thawing again, slow thawing or fast thawing again plays a very important role, because these when we are cooling, freezing the tissues, we are doing the natural vitrification. So if the cells are vitrified, and again when we are thawing, again we need to bring those particular cells or tissues in the vitrified stage. And then recovery condition also becomes very important. How we are trying to recover, what media particularly we are using, what conditions we are using, because the tissue has gone into the very big shock of desiccation as well as freezing. So this tissue needs a special type of recovery conditions and methodologies. So just again coming to the cryopreservation techniques, I will not be explaining much, because already I believe you have these in the earlier lectures, these techniques have been described. I will just go to the application part of these techniques on the non-orthodox seeds, like air desiccation, pre-growth desiccation, encapsulation, and vitrification. So this is the particular method which we generally use for developing the protocol for the embryonic axis. So this is almost same which is being used for the shoot apices. And but here we are going only for the air desiccation at the different period, and then putting the tissue in the cryo vials and plunging fast into the liquid nitrogen, that rapid thawing, and then going for the culturing. In the case of embryos or seed, we can straight away put the whole seed or embryo into the after desiccation, particular level of desiccation to liquid nitrogen, and then retrieve it and go for the regeneration. And this is the simple methodology which we use for the desiccation, freezing, and then putting those material into for the cryo banking. This is how we do the moisture content. Generally, we use these aluminum foil cups if the tissue is very small, especially in the case of embryonic axis. For the large type of material, we can use these moisture these vials which we can use small. There are different size of vials available. So depending on the tissue size, we can use these type of containers. So air desiccation freezing method for embryonic axis is this, and applying these techniques, we have cryopreserved successfully cryopreserved several species of citrus and underutilized fruits. This is the citrus ichengensis which I was discussing. This is the having the largest seed in the genus citrus. We could successfully grow these in vitro condition and desiccate them and regenerate. And this is in case of embryonic axis. Here you can see, and this is the case of whole seed. And we have applied air desiccation, vitrification, encapsulation, all these three techniques, and successful cryopreservation could be achieved. And similarly, in case of citrus megaloxycarpa, we have applied all these three techniques, and we could successfully cryopreserve them. In case of citrus indica also, this is the wild endemic and endangered species of Northeast, and we could successfully apply the cryopreservation techniques to seed embryos and as well as to embryonic axis. And healthy plants, we could transfer it to the pots also. And this is just a study which you will appreciate that for the different period of we have stored the germplasm. This is the study on 316 accessions of citrus embryos belonging to the 5 species, which were cryo stored up to 8 to 10 years. And we could successfully see that after cryobanking for these period, the original viability retained, or in some cases it increased little bit. So this is the published work in cryo letters. So in most of the cases, I should tell you, not only in citrus, in some of the orthodox seeds and non-orthodox seeds, we could find that original viability is retained. In when I talked the original viability at the time of cryopreservation, what was the viability? And after the cryo storage, when we retrieve it for some particular time, like few years, in some of the cases we have a cryopreserved material up to the 29 years now, and we have tested the viability, and it is very well retained. So this just to tell you. This is again some of the examples has been done at the large scale, and we have established the in gene bank the large number of materials, or in the gene bank, especially in the cordia and moringa and gigifus and several other species. So I will show you the status. In some of the citrus species, we could successfully preserve them, and we could get the very nice in vitro plantlets after cryopreservation. Here you can see these are all in vitro plantlets. In Bucrania, in Salvador also using embryonic axis, in Musa also. But this is not the case everywhere. In all the species, this is not the similar case. As I talked, in the highly recalcitrant species, still we are struggling. Like in Maduka, here you can see the case. We can get the survival, but there is no whole plant, we could not get it. Like in the same thing happened in the jackfruit, same thing happened in the lychee, and same thing happened in the jamun, in Shygesium. And lot of experimentation we have done during our projects and as well as our institutional projects, but still we are struggling to get the good success. In some of the cases you get the root but not the shoot. So why this is happening, we have actually studied using the ultrastructure of the tissue after desiccation as well as cryopreservation. This is the normal shoot apex. You can see the very healthy cells are there. Meristematic cells here you can see. But in after cryopreservation, you will find the cells are damaged, they are highly vacuolated, and so many other changes are happening into the cells. And those cells are unable to retrieve when we give them the those particular environment, and we want to regenerate them. Regenerate those cells. So there is a damage taking place in the subcellular organelles, which the cell is unable to recover. So this is the main thing which is happening. And for the highly recalcitrant species, what we have thought about is this is the alternative strategy is available, that field gene bank cryopreservation, vegetative tissues we can go, somatic embryos, and pollen cryopreservation. This is we have taken up in all the major recalcitrant species like mango, lychee, jackfruit, and others, where we are unable to cryopreserve or anything. We have gone for the pollen cryopreservation, and this is the successfully we could demonstrate in mango and lychee and other species. This is again another technique, regrowth dehydration. We are in this particular case, we are giving the treatment to the explant of cryoprotectants, and then we are going for the cryopreservation, especially in coconut. This technique was first utilized by Bajaj and Chin, and then Florent Engelmann and all. And this technique is later on shifted, applied to the other also. Like in vitrification, has already been explained in a few some lectures also. This technique is we are also applying on embryonic axis, especially using the PVS2. And this is the methodology which we are using, excising the embryonic axis and then going for the vitrification and plunging it into the liquid nitrogen and then retrieving using the in vitro methods. And using this vitrification technique in jackfruit embryonic axis, we could get some growth in the control or after desiccation, not after the freezing. And then encapsulation and dehydration is also one of the very good and common method for cryopreservation, which is we have applied on almost several embryonic axes of the different species, and successfully we could cryopreserve them, especially in the citrus and several other underutilized fruits. So this is one of the species that Ponceres trifoliata. We have applied all these three techniques. We had this air desiccation, freezing, vitrification, and encapsulation, and we could get the very good plantlets after regeneration. But in lychee, here you can see after vitrification, we could just get the growth in shoots, not in no roots. And here in encapsulation, we could get the good root, but not shoot. So these are the difficulties which are again and again we are facing. Even the application of these highly new basic techniques. Again, in lychee, a lot of cryopreservation experiments we have undertaken, but I should say that we could not get the very healthy plants. But in citrus, this is Citrus micropatera, we could get the very plantless. But in some of the species in Jambiri and other, we are also facing problem with regeneration after cryopreservation. So quickly, I will show you the cryo banking part, because you are not physically present in the NBPGR, most of you. And I thought that it will be good to little bit show you how we are doing the cryo banking after developing the cryo protocols, how we are moving ahead. So just it will give you the glimpse of the cryo gene bank and processing of the material and all. Quickly, I will go through some of the slides. So laboratory requirement for cryo banking and cryo storage of plant germplasm. These are the two things which are very important. And cryobank provides the long-term conservation of biological samples, safe storage for posterity, sizable infrastructure and recurring cost is involved, and if found unwanted and undesirable, because cryopreservation samples once preserved in the viability of the curation and the institution also. We have to very amicably plan what actually we want to go for the cryobanking, cryopreservation especially. And storage system once operational cannot be changed frequently. So we need to have the intense futuristic thinking before establishing any cryo lab or cryo bank. So this is a must. Then selection of inventory system. Actually, what we are going to cryopreserve: embryos, embryonic axis, shoot apices, and what other materials. Especially cryobanking is not only for the plant tissues. In biobanks, you might have heard of the name. And a lot of stem cells and organs and tissues are being nowadays cryopreserved, and there are several private companies have gone into this business in the biobanking, and they are having the good infrastructure developed in the different metros now. So we have to very carefully select the inventory, because especially when I talk of the plant tissues, the inventory should be very flexible, because we have to handle different type of material, like starting from the large sieve to small embryonic axis. So our inventory should be very flexible. Then data management is again very important of cryo stored accessions, and this facilitate the password data, physical positioning of that particular material. Physiological parameters are of immense importance, because all the material which is being cryopreserved, it is to be retrieved some or another time. So we have to have all data, all our experimental things, that how it is to be retrieved. So defining storage requirements, we have to see what material is to be stored, how many samples are to be stored. Inventory system is a must. As I told you, it should be canister or racks, sample to be stored in vapor phase or liquid. This is again important, what type of material we are preserving. We want it to put it in vapor phase at -160 to 196, 180 degrees Celsius, or in the pure liquid at -196. This we have to decide prior to the going for the experimentation and all. Then what will be the liquid source, bulk, LN station, small quantity, how much our target is. So that all these things we have to see. What temperature are required for protocol development, the storage systems, autofill or manual. So all these things we have to decide. So while deciding all these things, there are the four C's we have to look into: the cost, consumption, capacity, and control, temperature, autofill, and monitoring. So these all the things we have to decide. And nowadays there's another addition to the all cryobanking is the robotics. People, some of the countries have already the robotics facilities where no need to human intervention. Then the cryo containers, as I told, the microprocessor based, auto filling through solenoid valves, and generally all the cryo tanks which are coming nowadays, I have all these characteristics, and even some of the are having the very advanced characters. You need not physically present there. Once the material is put inside, there is auto filling, auto alarm. You can have the cameras and everything there. You can just see and monitor through your from the very distance also. So all these facilities are nowadays available. But for developing cryo protocols, you need a portable storage tanks or shipping containers. One are also important, and dry shippers. When you are developing the protocol in a different laboratory and want to bring that material to the cryo gene bank, these shipping containers or dry shippers are important. So just to show you the photograph, this is the LN vessel which we have at the NDPGR. This is a 6,000 liter capacity liquid nitrogen vessel, and these are the small tanks. These are the cryo tanks which we have in the cryo banks at NDPGR, but there are the different types are available nowadays. This I found the most convenient. I have seen the several other also in the cryo banks, but this particular cryo tank of MV is very comfortable to retrieve or place the material inside. And these are the dry shippers which we use for transporting the reserved material. So this is the how the this is the old cryo. This is already more than 20 years. This facility is created at NDPGR, and you can see the monitoring system and this microprocessor based and all. This is the vapor phase and liquid phase. These are the two just figures of two tanks. This is the vapor phase storage tank. Here you can see the platform. What we have in the cryo in the gene bank, in our cryo gene bank. These are this type of tanks are used for the when we preserve the material at vapor phase. And here we can see the complete liquid inside. This is the platform. Liquid will be above this. Material will be above this and liquid will be below this platform. So all the material is in the vapor phase. Here all the material is in the liquid phase. So depending on our requirement, we can choose the tanks. And there are some advantages and disadvantages of liquid phase and vapor phase. So I will not go into the detail due to the shortage of time. And sample storage containers, as I told, the most preferred ones are polypropylene screw cryo vials. And here also we need the inner thread cryo vials for cryobanking. These are the different types are available: 1 ml, 2 ml, different size, 5 ml, 50 ml are available. So depending on the size of the material, we can go. In Poland, we can just use the 1 ml or 2 ml cryo vials. So this is the inventory system, which is very flexible. You can see the different things. And here we, for the large seed, generally we use the polyolefin tubings also. Here polyolefin tubings or different diameters are available, which you can, these are the very shrinkable tubes. Through this dryer, you can shrink them, and these are the wooden caps which you can put inside, and you can just seal the material. So this is flexibility is also available if the material or germplasm is very large, especially in the dormant bud sticks. If you are preserving, or large seed if you are preserving, then polyolefin tubes you can use. And these are some of the basic equipment which are required for the small cryo lab. And then other specific laboratory requirements are seed germination testing and BODs, cryopreservation lab, is backed by the in vitro facility also. These are all things which we required while we want to establish the cryo lab. Safety consideration, because liquid nitrogen, when it will fall on your skin, it will burn it because the temperature is very low. So we have to take care. Oxygen monitorings and all these need to be placed, and closed circuit cameras and all. So coming to the cryostorage plant germplasm, briefly I will just try to explain how actually we are doing it. This is the flow of germplasm while when it is coming to cryo for the cryo banking. The plant germplasm, as you see, after collection or getting it procurement of germplasm, we need to study the morphology and physiology, then development of cryopreservation protocol, as I explained to you. And then if this is a small laboratory, the crop-based institute or something, then we go for the small scale laboratory, we develop the protocol, and then we go for the cryo storage. If we are doing like NBPGR, we have the large facility, large laboratory, then large scale laboratory at conservation mandated institute like NBPGR, then we go for the long-term conservation and cryobanking. From here in the small crop-based institute or small lab, the material can be shifted to cryobank. And these are some of the priorities. What is to be cryopreserved? This depends on each institution or each country. They all have set their priorities. Like basically, cryobanking is being used for non-orthodox seeds as well as vegetative tissues and pollen and several other type of material now are being cryopreserved. Even some of the recent publications have come that the orthodox seeds also are being they are advocating that this should be cryopreserved, like medicinal aromatic plants, wild species where seed is very less, registered germplasm, exotic germplasm, core collection and release varieties. All these are the priorities which have been set by the NBPGR, but each institute or each country can have their specific priorities. And the strategy for selecting suitable explant, it also depends on the desiccation and freezing levels. And what is our actually aim? We are going for the genotypic conservation, or we are going for the gene pool conservation. Depending on that, we can take the explant like embryonic axis, embryo, shoot apices, meristem, dormant bud, and several others. We can choose that. So this is just a summarized what I have already explained to you, and there are no international guidelines available for the minimum number of seed to be cryopreserved, or the minimum number of the explant to be cryopreserved, as these are available for the traditional gene bank. So 2000 seeds for self-pollinated and 4000 seeds for the cross-pollinated, but in cryobank there is no such guidelines as such available. But when we are preserving these, we want that the possibly large sample should be stored depending on the availability and also the reproductive biology of the species, as there would be greater chance that rare and potentially useful genes would be represented in the stored sample. In case of other explants, number usually depends on the availability of material. This is the constraint, and I should tell you for the especially when we are handling the tropical tree species, and percentage regrowth success and plan of retesting. So depending on all these factors, we need to decide the number of explant. And all the explants, when we are going for the base collection, the number of, if we are having the 100 explants to be preserved, we would try to preserve them in the 5 cryo vials or like. So all the material or the sample is preserved in the replications, not the all 100 to be preserved in the one, because once the thawed, the explants cannot be refrozen without the damage. So these are some of the care when we are doing the long-term cryobanking. We should keep all these things in the mind. Another part of the sample should be stored separately. A test sample we can store, and then for testing the particularly viability after 10 years or so, we can also do these type of things. So using all these techniques and cryobanking, I will just give you 2 examples. One is the of citrus, more than 971 accession belonging to more than 30 species from indigenous and exotic sources, cryo-stored in the form of seed, embryo, and embryonic axis. So these are the number of species. You can see the detail. These are already cryobanked in at the NBPGR. And similarly, there were around 1,200 accessions of several underutilized fruits in the form of embryonic and embryonic axis. We have cryostored successfully, and regularly we are retrieving and testing the viability in some of the cases. So this is the exactly the present status of the cryogene bank, where we are having total more than 12,000 accession now, and the number is increased because this is a little bit old data. And you can see the most of the material which we are targeting is fruit and nuts, spices, plantation crop, agroforestry, industrial plants, and medicinal aromatic plants. In orthodox seeds, special our emphasis is on wild and weedy species and crop wild relatives in millet, forage, pseudo cereals, grain legumes, oil seed, fibers, vegetables, medicinal aromatic plants, narcotics, and other some of the miscellaneous. And in dormant birds, we are especially preserved the mulberry and some other temperate fruits. Then pollen grains are of mostly of mango pollen. We have preserved lychee and other jackfruit and some of the other fruits. And genomic resources also we are cryopreserving. So this is the complete status of the cryogene bank at present. All the data which is being the material is cryopreserved is in the put in the cryogene bank database, which you can also access on our website. This is just the screenshot of that cryogene bank database. And some of the various publication, if you want to see something in the detail, you can refer to our publications. This is the one of the publication where totally on a practical approach, cryopreservation of plant species. And this is the recently published publication on management of citrus genetic resources, especially for exploration to conservation, and cryotechnics and all these have been described, which can be also implemented on other plant species of similar nature. So thank you very much. I hope I could do justice to my topic.
Thank you very much, sir, for a nice presentation. Friends, you can quickly ask any question or any query with, if you have, with our speaker. Dear participant.
Hello, sir. Hello.
Yes, yes, you are audible. You can ask.
सर मेरा क्वेश्चन ये था कि जैसे फ्रूट स्पीसीज है सर, जनरली उसमें रिकल्सिटेंट टाइप ऑफ सीड पाए जाते हैं। हेलो,
यस यस,
तो सर उसमें ये उसको प्रिजर्व करने के लिए मतलब वो क्रायो प्रिजर्वेशन में जाएंगे या इन सिचु करेंगे उसके वैरायटी या जल प्लांट को प्रिजर्व करना है तो?
एक्चुअली जो देयर आर सेवरल मेथड्स अवेलेबल, आपके पास डिफरेंट मेथड्स हैं, इन सिटू भी है, एक्स सिटू भी है, और जनरली जो अब आज लोग हम लोग जो प्रेफर करते हैं वो कहते हैं कॉम्प्लीमेंट्री कंजर्वेशन स्ट्रेटेजी। आप एक ही टेक्निक के लिए नहीं जाएं, बल्कि आप मल्टीपल टेक्निक के लिए जाएं।
ओके,
हाँ, जैसे फील्ड जीन बैंक है, वो भी एक मेथड है, इन सिटू कंजर्वेशन है, क्रायो बैंकिंग है। तो डिपेंडिंग ऑन द स्पीसीज उसको क्या रिक्वायरमेंट है, that method you have to use. You can even use one or two methods also for one species. No, I need not stick to the cryobanking or need not stick to the field gene bank. Both, because a lot of material which we have cryopreserved is from the field gene banks. So the field gene banks are highly vulnerable. क्योंकि especially in the case of fruits, आप लोग अगर कोई somebody is from the horticulture side and fruit side, वो उनको अच्छे से पता है कि कितनी field में. So that we are preserving in the cryo bank also. It will be just like a double safety.
Right, sir, right. लेकिन सर जैसे फ्रूट स्पीसीज में अगर क्रायो प्रिजर्व करते हैं तो उसका रीजनरेशन प्रोटोकॉल मैक्सिमम में एस्टेब्लिश है सर? अभी
आपको बहुत सारे एग्जांपल दिखाए हैं जिसमें कि
देखें जी
रीजनरेशन हम लोग इजीली कर पाए हैं, और इवन वी कैन गोस टू द फील्ड. Some of the material actually we are regularly giving to our crop-based institute and the field gene banks. Recently gave a lot of material to the PAU of Bhuvar. Dr. Subhash knows that.
Yes, yes, sir.
Okay, sir.
Recently, we have provided
Okay.
the citrus material to Abor regional station of PIU.
Okay, sir. Thank you. Thank you.
Any other question, friends?
Hello, sir. Hello.
Yes.
Please.
Sir, during 2 decades, I also visited your center, Abohar. It's very good center, sir. With
Yes.
Dr. Y.S. Ahlawat, sir, plant biologist, sir.
Yes.
During my PhD work, sir.
Okay. Very nice.
Thank you very much, sir. Thank you very much, sir.
Any other question, please? Friends, any question, any doubt? Even
Sir?
after this.
Yes, please. Boli.
Sir, one more question, sir. That we are having one differential calorie screening calorimetry. So how this can be
DSC
used
you are
in
having. You are having
Yes.
DSC.
Yes, sir. Yes, we have.
Okay. Very nice. So are you using that DSC?
No, sir. We want to know how it can be used in the cryopreservation studies.
This is the very important equipment for studying the developing, especially the cryo protocols for the difficult to store materials. And a lot of studies have been already published using the DSC. We also had the DSC, but for a long time we are not having because our major emphasis on the application part. Whatever protocols we are developing, we are applying those protocols to the germ plant. Being the NBPGR, our mandate is to long-term preservation of the material. But DSC is specially used for the basic studies. See the water content in the cells and outside the cells and intercellular spaces and all that, and how that is taking place within the different period while we are doing the desiccation and freezing. That data will be provided by the DSC. So I don't know from which company you have brought it. Perkin-Elmer and other companies are there, and they can still provide you the training if some of the person are not trained. But DSC is very important especially for the basic studies and during development of the status of water during freezing as well as during desiccation. So we can discuss it in detail later on.
Okay, sir.
Sir, one question from Muhammad Raja from Kazri.
Yes.
How many samples to be stored in cryotank in one time?
One time?
One time. यानी once. एक बार में हम कितना करेंगे? एक बार में in one time. Muhammad Raja, are you there? You can ask your query directly.
थोड़ा और explain कर दें क्या?
हाँ, आप क्या पूछना चाह रहे हैं?
सर नमस्ते। हाँ, मेरा वही क्वेश्चन है सर, एक ही टाइम पे हम कितने मतलब सैंपल स्टोर कर सकते हैं उसमें क्रायोटैंक में?
नहीं, क्रायोटैंक एक्चुअली डिफरेंट साइज का क्रायोटैंक होते हैं।
ओके सर,
कैपेसिटी अलग अलग होता है क्रायोटैंक का ना।
हाँ, कैपेसिटी पूछ रहा हूँ सर एक्चुअली ना।
हाँ, जैसे हमारा अभी क्रायोटैंक है उसमें हम लोग 4 से 5,000 सैंपल रख सकते हैं।
अच्छा।
और इसी तरह से कोई दूसरा टैंक है जो जिनका कैपेसिटी होगा 1000 का, किसी का 500 का, तो वो डिपेंड करता है।
ओके सर, डिफरेंट कैपेसिटी है मतलब
हाँ,
स्टोर
डिफरेंट कैपेसिटी के टैंक आते हैं और ये भी डिपेंड करता है कि आपका साइज कितना है जो एक्सप्लांट का। जैसे अगर आप कौन सा मटेरियल कर रहे हैं, पोलन कर रहे हैं या कोई सीड कर रहे हैं या एम्ब्रियोनिक एक्सेस कर रहे हैं या एम्ब्रियो कर रहे हैं या शूट अपाइसिस कर रहे हैं, तो जो साइज ऑफ एक्सप्लांट वुड आल्सो मैटर कि आप एक टैंक में कितने सैंपल स्टोर कर सकते हैं।
ओके।
जो स्टैंडर्ड टैंक आते हैं वो 2 ml या 1 ml क्रायो वाइल के हिसाब से आते हैं, क्योंकि वो जो जनरली जो ये जो क्रायो प्रिजर्वेशन है या क्रायो बैंकिंग लार्ज स्केल है, वो मेडिकल साइंसेज में और एनिमल साइंसेज में यूज हो रही है। प्लांट साइंसेज में तो ये 1970 के आसपास से ये आई है। तो स्पेशली जो टैंक्स और ये जो इक्विपमेंट जो बनाए गए हैं, ये मेडिकल साइंसेज और प्लांट और एनिमल साइंसेज या वेटनरी साइंसेज को ध्यान में रख के बनाए गए हैं। तो प्लांट्स प्रिजर्वेशन के हिसाब से बहुत ही कम अब बनाने लगे हैं कुछ लोग, पहले ये नहीं आते। अभी भी ये ड्यूअल टाइप है, तो वो है इनमें इश्यूज, क्योंकि हमें अगर जरूरत है प्लांट टिश्यू के लिए तो हम लोग बड़ी फ्लेक्सिबल इन्वेंटरी प्रेफर करते हैं, जैसे आपने देखा।
जी सर, ठीक है सर, थैंक यू सो मच।
एनी अदर क्वेश्चन? Friends, if there are no question or you would have another means question after that, even you can ask from our speaker through email. We will share the email of sir, and then if there are no question, then I may propose a formal vote of thanks, sir. With your permission, sir.
Thank you, bhai. Thank you.
I, on my behalf and behalf of organizing committee, I thank you very much, sir, for accepting to deliver a talk despite your busy schedule. And today you share your knowledge on conservation of non-orthodox seeded germplasm using cryopreservation techniques. Sir, certainly our participants are benefited by your highly informative lecture, which you covered many aspects of cryopreservation, including exploration, laboratory handling, different techniques for cryopreservation, and experimentation with suitable examples. And you shown, sir, beautiful photographs. And finally, cryobanking, how to perform this on a non-orthodox seed species. Thank you very much, sir, again for accepting our invitation.
Thank you. Thanks to organizers and all the participants for patience listening. Thank you.
Sir. Thank you very much,
Thank
sir.
you very much, sir.
Thank you.
Okay, sir. Friends, afternoon at 2:30 we will meet, and Dr. Jyoti will take up then. Okay. We'll see at 2:20.
ठीक है।
अच्छे से खाएंगे। अभी ये रोटी मतलब ना मैम।
No, good afternoon. Good afternoon, Dr. Uttam. Sir, you are muted.
Good afternoon, madam.
Sorry. Good afternoon, everyone. Sir, you can hear me? Jyoti madam, you can hear me?
Yes, sir. Yes, sir.
Okay. Thank you very much, sir.
So how are you, sir?
I'm fine, thank you.
Okay. So this is sir second last day of the training program.
Yeah.
So it was 1st to 21st. So tomorrow is the last day. So tomorrow we'll be closing the day. Now only a few lectures are left. So as you were busy in the previous week, so that's why we today. So before that we had on Saturday lectures on particularly avitic stresses and those things by Dr. Bara Prasad, Dr. Viswanathan, Dr. Jagdish Rane.
Oh, excellent.
Then today Dr. Rabi Sahu.
You're right.
He also delivered drone phenotyping, so remote sensing and
Excellent.
so now it's your turn. So just I will brief about the trainees. So here in this training, sir, we have trainees from different backgrounds. So mostly they are from genetics breeding only, breeding backgrounds, working on different crops like cereal, legumes, oil seeds, then tree species, and they are from different institutes, SAUs, ICR, some private institute. From CG also ICARDA too are there. And most of them are like assistant professor or scientist level, around 45 or 50 of them. And some are principal scientist or senior scientist, like that. So good afternoon, one and all. So good afternoon, dear participants. So today our speaker is Dr. Uttam Kumar. So I will just introduce about him. I will share my slide. One small slide. Just a minute, sir. I will.
Yep.
So whether the slide is visible?
Yeah, okay. Okay, yeah.
Okay. So again, good afternoon to one and all. So today's we have the speaker with us, Dr. Uttam Kumar. He's at present senior scientist wheat breeder at CIMIT Borlong Institute for South Asia, BISA, Ludhiana. He's posted since 2014 there. Before that, regarding his education, he completed his master's from CCS University, Meerut. Then he obtained his PhD from Banaras Hindu University, Varanasi. He is dad Lebanese fellow. Then he continued in TERI and he is fellow of TERI also. He did several postdoctoral fellow also before moving to CIMIT and BISA. So regarding his nature of research work and expertise, so sir has a great expertise in precision phenotyping, then designing of field experiments, participatory plant breeding, maintenance breeding, So he's looking after all the 3 big farm in Ludhiana, Jabalpur, and Pusa under the BISA in India. So he's the great support for South Asia program, particularly all the Indian program and Southeast Asian program. He's managing all the big trials and mega trials across the locations. So he has more than 14 years experience in wheat breeding for biotic and abiotic stresses, then abiotic stresses, then breeding for industrial quality wheat. Then he's also have expertise in molecular breeding, particularly for genomic selection, gene mapping, and expression. So he's greatly involved in all the phenotyping works and integrated with the molecular breeding also. He has been the project lead for more than 9 projects, including Bill Melinda Gates Foundation project also. Then he is the adjunct faculty in PAU Ludhiana for biotic and genomic selection aspects. Then regarding his teaching and guidance, so he has guided 8 PhD and 1 MSc students. And now coming to the publications, he has published more than 60 international publications with the high impact factor, and he has reviewed very good articles in more than 30 articles in different international journals of high repute. So sir, we welcome you all for this training program, and please deliver your lecture. Certainly your talk will be enjoyed by our trainees who are very much enthusiastic to learn you, sir. Okay.
Thank you very much, Dr. Jyoti, for a lovely introduction. So my screen is visible?
Yes, sir. Yes, sir. Visible.
Okay, yeah, great. So today I will go through, I don't know, always I wish I could deliver a presentation or the lecture physically, that could be like more interactive, but anyway, it's a virtual training and we need to move on with the science and with the time. So today I will give you some like insight on high throughput phenotyping that we are doing, and that can eventually be used, especially in context of like germplasm management, I would say. No. Okay, so my full screen is visible or only the small screen?
Visual.
Okay. Okay, great. So this is just one slide in brief. This is for those who don't know much about BISA. So BISA is like one of the international research institute which has kind of UN privilege given by Government of India, but managed by CIMIT. So there are 3 different locations on the research farm that we have in India, which has been strategically chosen by some wise policymakers or scientists. For example, Ladwal, one Ludhiana, Punjab, it represents one of the mega environment wheat, for wheat mega environment. Another is Jabalpur, central zone. All of you are very much aware, which is known for good quality, heat stress is there. Pusa, hot and humid. Some foliar disease comes there, no, and that represent part of Trai of Nepal, part of Bangladesh, no. Jabalpur for the settler zone, Ludhiana represent like mega environment as I mentioned, and it covers part of Pakistan, some part of Afghanistan, some part of Nepal also. Anyway. Now we come to the like main question. First question for understanding, what do you mean by HTP or high throughput phenotyping? Does it mean just use of sensor or just scale out? So to make it is, it's both. It's use of sensor as well as it's a scale out. So eventually what we can do manually, we have certain limitations. When we go for the high throughput phenotyping, means already we are talking about at a large scale. Now question is, what are those tools and technology that can be covered under high throughput phenotyping and that can be used to basically evaluate a large number of genotypes? Doesn't matter these are the advanced breeding lines or the pre-breeding lines or the germplasm lines. Irrespective of that, this technology can be applied everywhere. Now I further simplified why we need to use high throughput phenotyping tools. So there are certain points I made. For example, if we want to collect data from large number of plots, As I mentioned, human has its limitations. Suppose I want to take a note, let us say from heading or height, I can cover maybe 100, 200, 1,500 plot, not more than 1,000 in a day. But if we are using STP tools, we can cover several thousand in a day, or maybe in a half an hour. Or maybe in future, we have technology that can collect that same amount of data maybe in 10 minutes or 5 minutes. Anyway, future will tell about that. So measurement of secondary traits, which are correlated with the yield, that can be measured using high throughput phenotyping tools. These are called as secondary traits. But decisions are made based on the primary traits. So you will learn in a bit later in my follow presentation. For the trait which are difficult, for example, root characters. So if we do it manually, for example, need to uproot the plant, clean it, remove the soils, put in the special chamber, take the 3D image, analyze it. So that's complicated. But if there is any indirect way to measure the root rates, that could be very much helpful, and we can cover large number of plots. So it's a kind of non-destructive method. For example, it's mainly image-based or sensor-based data collection. We don't need to destroy or destruct the plant. So we can minimize the experimental error. Of course, suppose a person is taking versus data is being taken by the sensor. So there is kind of biasness can be removed if data is being collected by the sensor, provided that it has been properly optimized. For example, canopy temperature or physiological traits. If we collect manually, it may take a lot of time. But with STP tools, it can be done in a very limited time manner. And of course, when we collect data manually, we cannot control environment. Of course, STP also cannot control the environment. But what can be done? We squeeze the time limit to collect the trait or a particular data. So the environment fluctuations can be taken care. Minimize human error, as I mentioned. Save time and resources. This is one of the key factors why we want to implement high throughput phenotyping tools, and it can be automated. That's another interesting part, and it can handle large set of data with minimal effort. And of course, some of the people are from the genetic background and from the breeding. It's a kind of breeding is kind of number game. You have a large number of population, you have chances to get the extreme with the desired trait. So certainly it has a positive impact on the increased genetic gain. Yeah, I think I already covered save time and resources. It's a highly cost effective way as compared to the manual data collection. So there are some of the examples I will go through that we are routinely using in our breeding program. So the first and simplest one is the digital data collection. Of course, many people are still taking the notes in the field book with the pen, pencil, take the note in the register. But what we are doing since 2014, we are completely switched to this field book app. So we take the notebook in the field, collect the data in real time. So there are 2-3 advantages of collecting this. Number one, it's less prone to the errors. For example, someone is saying 23.3. So maybe you by mistake put 2.33. So it immediately flag the issue. Okay, no, something is wrong. So that can be corrected like in a real time, staying in the field. If you find out such kind of issue, maybe after the crop season, then you don't have the plant and you already lose the data from that particular entry. Anyway, this is just one example. Second is it takes care of the phonic sound. Suppose someone is speaking in a different dialect, some people understand in different dialect. So sometimes we prone to enter different value if we are taking note in the register. Third thing is the checks of balance. For example, your field team or your field supervisor, whether they are actually taking data from the field or from the actual field you want, or it's from the different plot. So all the values are GPS tagged. There is a feature in the app. All the values are GPS tagged, which are all the logged. You have the time log, you have the GPS tag, and you have the person name of the person. So all the information is kind of checks and balances are there. So it will ensure data has been collected at what time, at what location, and who collected, and how it was collected. And on top of it, once you are done, immediately you can export the data to the computer through different means. For example, you can send by email, you can send by WhatsApp, you can copy paste. A number of feature options are there to send data from tab to the field. and there is no need to save the data. It's automatically saved in the program and you export it in a real time. So it means you collect the data and same day you can do the analysis. And when we talk about the features, there are any type of trait you can imagine. It may be numerical value, it may be true, false, it may be date, it may be picture, it's a disease score, text, anything you can imagine can be recorded. That kind of flexibility we have in this field vocab. Now, the second thing is kind of hardware we are using. It's a PhenoCart. It's one of the simplest version of collecting the data. For example, PhenoCart is conceptual wise the sensor you are carrying in your hand. Instead of holding in your hand, you are just holding some kind of attachment. This we call as a PhenoCart. It has kind of self-propelled wheels and attached to several sensors. For example, the green seeker, DSLR camera, GPS, computer, you can even attach kind of USB or HD camera to record a video of the plot. If you want to look later how the plot looks like after the season. And this is just one kind of simple example. For example, I mentioned that NDVI sensor, canopy temperature, or infrared thermometer attached to this. So once you run the PhenoCart in the field, because it has the GPS, so it doesn't matter how you move. So already you pass through the plot, and GPS information are logged against every data points, and all the sensors are connected through a special software that's freely available. I mean, you can download. We can share that one if someone is interested to use. So all the sensors are connected to each other through a software, and all the information are collected in real time. And this is the information we generate. For example, based on the GPS logs and the value of the particular trait and the time. So we extract this information using a simple R script. So left side is like a canopy temperature that you see, and then on the right side is an NDVI value that we extracted from the PhenoCart. So these kind of just follow-up steps. So we assign the canopy temperature and NDVI value, whatever the trait is, to each plot. So here we just extract the information. Here we assign the information to each plot. Suppose I know the plot number 20, which is like let us say it's on X and Y coordinate. It's on a grid. It's a 2-1. So we get the value of the 2-1 plot. It's plot number 20, for example. We know what is the canopy temperature of that. We know what is the NDVI value of that. So based on that grid, we extract the information assigned to each plot. So this is just an example of NDVI that we have successfully done, or we are basically routinely doing this. Now, next is UAV. Frequently it's known as drone, and now these days it's commonly being used in agriculture. And just one update to the best of my knowledge. 4 or 5 years ago, there was a market of around 8-9 crore for the drone. But now I heard it's around 1700 crore. That has been jumped from 8-9 to 1700. That kind of progress is being made or progressing in this direction. For example, agriculture spray drones, and government has implemented some policy. So I appreciate that. Make in India. So we cannot import drone from outside. I mean, for the research institute, it's a flexibility, but for the commercial purpose, you cannot import from outside. So this is basically helping to develop in-house capacity. That's why this market has been grown significantly in last several years, from 700 to 1700 crores. The business. So. But you need to fly the drone. This looks a simple technology, but sometimes people think, no, it's very complicated. How will fly? It needs certain skill, technology, knowledge. But that's not the case. So once you know some basic information, so how to fly it, how to use it, then you are good to go. For example, if someone really interested to fly drone, I don't think it's a matter of like it's not a matter of like more than 1 day to learn how to fly it. For example, there is one kind of freely available software or app. It's a mission planner. Basically, you can make a plan. You decide at what altitude you want to fly, what is the speed, what type of camera you have, whether you want to do it for spray, what type of overlap you want to have. If you have this kind of basic information, you can make a flight plan, and in the end there is just a kind of triangle. You hit that button and it does the job pretty automatically. You don't need a special skill basically to maneuver the drone. You don't need that. Once it's done, cycle is completed, automatically it come back to, we call RTR, return to the launch site. And we need to take some care when we collect the scientific data. For example, we need to have the ground truth control points. So when we fly the drone, this is just one example from PUSA from this season. We have, this is like one of the major experiment with different treatment with the nitrogen fertilizer treatment. So we have these ground truth points. We just fly the drone as per the paths defined, take the picture. So this is just one picture, but like this we have thousands and thousands of pictures, and we need to stitch them together. To stitch the image together, we need to have this precise information. This we call ground truth points. It helps to stitch the images which are like from the neighboring plot. And of course, when we want to extract the information from each plot, then we need this information. So that is one kind of requirement. We have the ground truth points, but for agriculture, spray drones or purpose just to spray, we don't need this kind of information. That is a difference. And this is just one example. No, for example, it was like normal RGB, we call 4K camera. We took these pictures, and this is the picture I just copied from the computer folder. So how the images are collected. For example, it's from the multispectral camera, which has 5 different bands. Like if you see the image name, if it's visible, 600-1 _1 _2 3 4 5 like this. So this is one image basically. The 5 images are from 5 different bands. We call this as a image 1. And then it's 601_1 601_2. So they are different spectrum of the camera. So if you want to make a RGB image, as you know, red, green, blue, infrared, and near infrared, these are 5 bands. So using these RGB images, we can constitute like a colorful image like this. We call it as a orthomosaic. I will show some of the plot slide. So this is how it looks like when we collect the data, and we don't get any information visually unless we really stitch the image together, make orthomosaic, and analyze it. And this is just one example. I think most of you know that point and shoot camera for NDVI. So what we have done, there are these are called kind of passive sensor. I think I would like to mention briefly the active sensor and passive sensor. The handheld green seeker, it's basically an active sensor. It has its own source of light. It throws the light on the plant canopy, and the reflectance is absorbed back by the sensor. That is an active sensor. But this camera, they don't throw any light. They are dependent on the ambient light that's coming from the sun. And every time we fly such kind of passive sensor, we have to have a calibration panel, and we take the picture. That is called as a reference panel. So every time you fly, irrespective of the daylight, that reference panel is work as a reference point. If sunlight is more, then value of the overall plot would be more. Sunlight is less, value for the overall plot would be less. So this is the purpose. So what we have done, we made a small modification. This camera, this is one of the low-cost sensor. Basically, we remove infrared blocking filter and add the red light blocking filter. So this normal camera can be used as an NDVI camera, basically. And when
so here, how the value will come? NDVI value?
Okay, so basically what we do, ma'am, this refractive or calibration panel, we take the value of the calibration panel every time. Oh, sorry, we calibrate once, and every time we process the data, the image is along the plot is there. So based on that original calibration value and the calibration value at that time point of that panel, so these both points work as a reference point. So any plot which has like a higher absorbance of the camera or low absorbance of the light, so it basically based on that difference extract the NDVI value. So it's a very simple process.
Calibration of image means reference point is like image.
Yes. If you see here, like we have the passive, infrared blocking filter is removed in the camera, and red light blocking filter is installed. That don't allow to pass the red light. Okay, because you want to record the greenness. So more green, more light, green light will go to the sensor, and then less greenness, less green light will go to the sensor, red light will not be there. So this is just kind of low cost, but we can go with the, let us say with this multispectral camera. It has already 5 different camera with different spectrum.
Okay.
So this is just an example, you know. The below, this is the orthomosaic prepared by this normal camera. So this is like one of the 3-4 years ago. I deliberately put this picture, which has been taken 3-4 years ago from Ludhiana. So we prepared the orthomosaic using this camera, if you can see on my screen, and with the same camera, of course, since it's we made this RGB picture also, but the same camera was not here. It was a different camera. Basically, it was a multispectral camera. So using these R, G, and B 3 camera we used to compile collate this one, and it is a simple procedure. There are scripts available in the R program that can basically extract this information. I will go through briefly to that one. Anyway, so this is just a result file. So NDY data that we collected through UAV. So here we can plot. There are different ways to represent how we collect the data. So this is just to kind of see the quality check after collecting the data, to see whether everything is in order or not. Anyway, so now someone asked the question, what other type of data that we can extract using UAV? For example, one is the NDY data. If we have, let us say, thermal camera installed at the UAV, which is a bit heavy, that's not being used at the moment by us, because the drone we are using, it's really very lightweight. Canopy temperature can also be extracted. Lodging information, yes, can be extracted. Plant height information can be extracted, and visual orthomosaic can also be made, as I showed this green picture in the previous slide. And this is just brief. Basically, it need a complete 3 or 4 day training program, basically, to learn how we extract the information from UAV or from the image data. So just to show you that first step is to make a orthomosaic. And orthomosaic is being made through Agisoft software, Metashape software. So we load all the images from all the bands, and we can prepare the orthomosaic. And there are several packages. For example, this is one step to make the orthomosaic. And next step is to extract the information from that orthomosaic. So based on the R packages, R statistical software, and we need to load a certain library, and there is some known already written scripts are there. So this is just an example. I cut it to the short, that it's not a complete script, to show that we can extract the information using known scripts. My purpose is to show that known scripts are there. You don't need to do much effort, only change the folder names and as per your requirement. And this is again several packages. For example, what we did, we collect the data using UAV, and then we collect the data manually, and we compare both the methods, whether they are correlating well or not. So using several known scripts. And this is one important like outcome of that exercise. So we compare the like UAV based data and the handheld sensor based data. We call like green seeker based data, and we found very good correlation. For example, it's not necessary that at all the date it will correlate, but still you see the correlation is quite high at different dates. So for example, this is the data from 2016. Still I have this old table, but I should update the new one anyway. So you know, we have a very quite high correlation between the UAV data and what we get with the handheld sensor. That is the purpose to show here. So okay, as I was mentioning, what are other extra data that we can extract using a UAV? For example, the NDY value I mentioned in detail that we can extract using UAV. So this is just one calibrated orthomosaic. Calibrated orthomosaic means when we fly the drone, we have this kind of ground truth control point and the calibration panel, and then we extract the information from information of each plot that has been assigned. So, for example, we need to remove some effect. If you take the data, there is some vegetation or some soil background is there that need to be removed, and in the processing step, we can take care of this step. And a second important part is, you just imagine, you know, when drone is being flown, it's taking picture where let us say with 75% overlap or 80% overlap, so like a side and forward. And if there is overlap, of course, it's taking the picture with an angle. Once you take a picture of one spot from different angle, it's possible to make a 3D image of that. So based on that 3D image, we prepare kind of a digital elevation map. We call like a digital elevation model has been prepared for that. So once you have the digital elevation model, you can extract the plant height. And once you extract the plant height, you can extract information about the lodging also, how much lodging has happened to that particular plot and what is the actual height of those particular entry. Because our plot is basically GPS mapped, as I mentioned, using these ground truth points. In reference to those, we make a grid and we can extract the information from each and every plot of our interest using the GPS coordinates from the corner of each individual plot. And this is just an example to show whether we are going in the right direction or not. If we extract the data using a drone or we have the like actual plant height measure, and you see that there is a very nice correlation between the data extracted using collected using UAV and the data collected actually using the scale in the field. And sometimes it needs to be very clear whether we want to work with the absolute plant height or want to work with the relative plant height. For example, initially we started with the relative plant height. For example, the red is the taller plant and blue is the let us say dwarf plot. So and the rest is kind of range. So this was kind of a relative information. But if you want to get actually absolute value, whether plant is 90 centimeter or 100 centimeter, that's also possible to extract using this digital elevation model. And this is an example, the NDVI correlation with the ground-based tool. So we still we found very good correlation in the NDVI value that we collected using UAV and collected using handheld based or ground-based sensor. This is the same thing. Now, this is one of the digital elevation model where we use successfully to measure the lodging, how much lodging might have happened in a particular plot. So what we did, we conducted one experiment where artificial lodging was done with different degree, let us say 0% lodging, 25% lodging, or 70% lodging, or 100% lodging. So as you know, if this is kind of, this is ground level basically. If there is lodging, the plant would be more toward the ground. If there is no lodging, they would be like away from the top of canopy, away from the ground. So based on this model, we are able to basically extract the information how much lodging happened to a particular plot, which is very difficult and tedious part. Basically, when you do the same exercise manually with your own eyes, then you have to be like quite experienced person for that. To pinpoint it, but if you are taking help of the sensors, it's pretty accurate. And I think you have seen this is the newspaper from 4 or 5 years old, I guess. That time the lodging, the lodging happened at large scale. The end season rain and storm was there, and the problem was government doesn't have a mechanism to go to each and every plot and say how much lodging might have happened to each plot. And please don't share with everyone. My father, basically, I belong to a farmer family. My father didn't even plant wheat in one of the plot, and he got the compensation. So sorry, this is of the report. I mean, this is kind of, I want to pinpoint. It's not that he got like 500, 600 rupees. That's the point. Point is, we don't have a foolproof mechanism that benefit goes only to the farmer who suffer the loss. But if we use such kind of, satellite cannot capture that. Basically, it's very difficult to capture if lodging happened or not. Of course, the resolution of the satellite is still at the scale of, to the best of my knowledge, around 3 or 3.5 meter per pixel. That is the resolution of the satellite. With that resolution, still it's difficult to pinpoint how much lodging might have happened. But if you use such kind of technology like flying UAV over the farmer field, using simple model, we can estimate how much lodging might happen and the loss might have occurred to individual farmer, but that needs to be scaled out and tested at the pilot basis. So anyway, that's my purpose to mention that based on digital elevation model and using UAV or the technology, it's possible to measure such kind of trait or study such kind of impact of end season rains or something happened to the flow. And this is just one example to show the level of confidence whether our data information generated in digital elevation model is correct or not. So what happens, this is like a full-fledged standing plot, and this is like after lodging. And there is a model, we extract the information also, we subtract the value from the standing to the lodged one, and we can get to know how much lodging might have happened in each plot. This is just one kind of result I want to show you. And we got very good relationship. You see the graph on the bottom, we got kind of nearly linear relationship between the data that has been collected, basically the ground truth data and the UAV data. And not only for this measuring the NDVI and canopy temperature and lodging and digitization, not only that, beyond that, when we talk about up to what extent we can go using the STP tools. So for example, this is one of the study we have conducted. We use the sensor to do even the QTL mapping for spot blow disease in wheat. And in summary, I want to show it's very much possible. So there is much less biasness if we are going for sensor. So if you see these graphs, one is like manual distribution, the manual as well as the NDVI based distribution of the lines. And interestingly, you will see here the QTL that we measured using the conventional method exactly in the mapped in the same place where we in the same place when we use this NDVI or the handheld sensor. So it means this sensor can be effectively used for molecular studies also. And I think, okay, so probably I don't know if this can play from here. But there is a post-harvest data collection using open source digital seed counter. Any lab or any institute organization, if they have a simple DSLR camera or a good resolution point and shoot camera, they have the seed counter, it means in their lab. It's very simple. Just take the picture, have a uniform light on the seed, spread on the piece of chart paper. Take the picture, and with simple analysis, you can count grains. It's very accurate and fast. I don't know if it I'm able to play. Oh no, looks complicated, I guess. Anyway, so I can share you, the link is here. I can share this presentation with Dr. Jyoti. She is free to share with anyone who is interested to go through this. So here, so what we this is just a result, whether we count manually or we count using this image based processing, the 1000 grain weight. And we found some improvement if we use this image processing based counter compared to the manual. So manual also not bad, the heritability is quite high, but very similar. So these are the results from our grain counter, which is like low cost. Anyway, so I'm very end to my presentation if I'm not too fast. So I would say high throughput phenotyping is a necessity in modern breeding program. As you know that as mentioned earlier, breeding is a number game. Larger the number of entries, you have the chances to have the extremes. And for that, manually would be difficult, but if you implement STP tools, then it would be quite easy. And highly effective for large scale field testing. Of course, that's very well logical or self understood. So possibility to increase number of entries to be tested without increasing much of the cost. So only the field cost, whatever the trial management cost is there, but taking data from 10,000 plot or 20,000 plot doesn't make much difference in terms of cost. And it has been well proven that STP is efficient and can be used not only for selection, but for the gene mapping activity or work also. So in the end, I would like to thank all the collaborators. Some of are already missing. I didn't pay much attention to the last slide, but that's very important. For example, recently we got some support from DBT, Government of India, CRP, GIZ, DMZ, several donors are not missing, and the entire team who is involved in the work, and we are getting support from KSU, let us say, for this Fieldbook app and some of the Scripts got from Jesse Poland, the Finocart, basically supported by Trevor Reef, and some analysis by Jared Wang, and of course our CIMI team, Dr. Ravi Singh, Suchasmita Mandal, Dr. Joshi, Manish Pradeep, and our field supervisor. And thank you everyone. Thank you. Thanks for your attention. So if you have any question, please feel free to ask. Yeah, so Dr. Jyoti.
Hello. Yes, Doctor. Hello. Sir, your voice is not, Dr. Uttam, your voice is not coming.
Okay, now you can hear me. Looks like.
Yeah. Now it is. Yes. So it's over now, or means you wanted to?
No, I mean, if someone has any question, please feel free. Anything, you know.
Okay. Okay.
Yeah, but look like it's abruptly my screen share, everything is stopped.
Yeah. It looks like that it stopped abruptly. So that's why I thought that means you will be sharing again.
No, I don't know. I think this you can see. This is what.
Okay. So some dear participant, you can put the questions in chat box or you can ask directly. So now it is open for discussion. In chat, some question is there.
Okay, let me see how to go there. Okay. Will you please elaborate NDVI sensor procedure method for QTL mapping? Okay. Yeah. Okay. So what we did basically, we use this handheld NDVI sensor to measure, to go through each plot. But what happened, this is the experiment that we conducted in Pusa. We inoculate the field artificially. There was sufficient disease pressure. And the good line, the resistant line, they were still green, and the susceptible were showing kind of more senescence. And with the NDVI, it was able to capture precisely more disease, like more senescence, that was a higher value, less disease, more greenness, that had like, okay, of course, that has a like higher NDVI value, and disease plant has a lower NDVI value. So based on that, we conduct the QTL mapping analysis, and of course, we collect the data manually also. So there was significantly higher correlation. If you see in one of my slide here, okay, this showed kind of correlation on right hand side. The it was 0.83 coefficient of determination, right? R square, and the same for disease severity in germplasm line also. Here you are the mapping population on the top right side, and germplasm on the bottom right side. The correlation was very high. The manual data collection restoring and the data that we collected using NDVI. So this is what I mean we observed, and we are trying for yellow rust also this time. Let us say if it works, for example, we have very good disease pressure comes because of the weather this time. It was more favorable for yellow rust. We can see if same approach can be used for that, for yellow rust. Yeah.
Okay, thank you, sir.
More questions? I don't know.
Thank you, sir.
Welcome.
Thank you, sir.
Yeah, thank you. Welcome. There is no more question in the chat. I cannot see.
Okay. Yeah, please put the question if you have any, or you can ask further through mail also. So we'll be circulating email ID of the resource person. So in that, through that also you can ask. So about the like these are I mean affordable phenotyping, low cost phenotyping. Means Dr. Uttam has dealt with means all the fields, and very good phenotyping work has been being done at all the centers of BISA. And this NDVI is correlated with Rust response also.
I mean, yes, yeah, that's correlated with the Rust also, and we are trying this year to see how it goes. I mean, we didn't try earlier, especially for the Rust. We can try this year, and I can share the link of my presentation if someone is interested to go through. Give me a second.
There is one more question in chat, Dr. Uttam.
Okay, what is this? Digital elevation map you show can we use for precision agriculture? Means you can check drought heat. Okay, yeah. So digital elevation model, like it's a simple concept basically. We have the ground truth control point. We know the GPS point as well as the elevation of those points. And when we fly the drone, we have the top of canopy also. So based on that, we can actually see how much is the height of a particular plot. Just imagine, since we are collecting data from different angles of the same plot. So once you have data from different angles, you can make kind of 3D model. So this combination of 3D plus a digital elevation model, so you can pinpoint precisely what is the height of a particular plot. So this is what we can do. And when we talk about to measure drought or heat through this, of course, let us say canopy temperature. This is one of the like parameter. Basically, the plant which tend to have the cooler canopy, they tend to have more yield. So that is one indirect way. But still, there is no guarantee whether it would be tolerant to heat. But still, we can make a best guess what could have been. I mean, which line could have been more tolerant or resilient to heat, and yeah. And of course, there are certain sensors. If we use, we call it. It's a complete spectrum of complete spectrum of image. How to say? No, with how to say with the hyperspectral camera, there are several bandwidth that can tell or give you an idea about drought and no drought. Yeah, of course, response of plant, response of leaves, or I don't know, no, how to say, reflectance of the leaf in response to the drought or heat. That changes basically. So with that complete spectrum, we can get to know which specific spectrum is pinpointing the response to the stress. It may be heat or it may be drought, but seems to be more feasible for the drought rather than heat, because heat is again measured purely indirectly. This is what we are trying, but that precise answer could be given maybe in a couple of years to learn more about that. But it's possible using using UAV. But on top of it, when we talk about disease or biotic stresses, that's relatively easier to find. Even using hyperspectral camera, it's relatively easy to the best of my knowledge as compared to heat and drought.
Means a hyperspectral camera is better than multispectral.
I mean, it's not that you always use hyperspectral camera. So what happens? Hyperspectral camera has a complete range. Let us say from 300 nm to 2400 nm, and you need to basically find out which spectrum is related to which stress. So some spectrum variations come in response to drought. Some spectrum variations comes in response to some disease. Some spectrum come in response to, let us say, heat. So once you find out, let us say, once I know this spectrum, let us say 950 is pinpointing our drought stress. So you don't need to use the hyperspectral for that. Use a like single bandwidth spectrum camera that could be low cost. So this is how it works. Otherwise, hyperspectral, I mean, we are trying to conquer. I think that's quite expensive. I don't know, in Indian rupees should be 1.2 crore to 1.8 crore. if you want to buy a good hyperspectral camera?
Okay, so it depends upon that.
Yeah.
In software, for all the spectrum bands, that modeling data is available for different data points and the spectral data. Means
No.
conversion, I am asking about conversion of suppose NDVI to yield or NDVI to disease resistance. So
Yeah, I mean some parameters are simple to convert, some are complicated to convert. But I don't know, only the people maybe a little bit from the physics background and who are really working. I know some people are working on the hyperspectral camera. So just imagine, hyperspectral is many times complicated than the normal RGB camera. So what you see in the normal RGB camera is just a kind of two-dimensional picture you see, which has length and width. But when we talk about a hyperspectral camera, it has basically length, width, and of course height. So it means, let us say, if I have one set of images, basically there are thousands and thousands of images to my understanding. So I think those people know better. It's very like computational intensive to get information from those from hyperspectral camera. It's much easy to analyze using single band multispectral or RGB camera. It's much easy to analyze.
okay. Okay, so if there is no question now, then I would like to thank Dr. Uttam on my behalf and on behalf of organizing team of this virtual training and on behalf of Director NBPGR. So we thank you very much for accepting our invitation and delivering such a nice lecture on the high throughput phenotyping for large scale evaluation. So thank you very much, Dr. Uttam. Yeah, we'll be sharing email ID of Dr. Uttam to all the resource person. We'll be sharing a compendium where all the details will be there. So, so you can ask further him about the. You have already said, okay. So he has already said. So
yeah, this is my presentation. Anyone can download it.
Okay. Thank you, Dr. Uttam, for sharing here. So, thank you very much.
Thank you, Dr. Jyoti. I also would like to thank for providing me this opportunity and sharing what we are doing with the rest of the scientist colleagues.
Yeah, surely. So all the participants must have enjoyed the lectures. They must have gained the knowledge, and they may implement in their research work further in the future. We hope so. So, so thank you very much for imparting such a useful talk.
Good, yeah, thanks. Yeah, so I'm allowed to leave or?
Yes, yes, you can leave.
Okay, thank you very much. Thank you.
Okay.
Ma'am, 3:30, ma'am, actually, no?
Okay. Okay, dear participants. So our next lecture is at 3:30. So that is a very important lecture about how to know the registration process of genetic stock for plant germplasm registration committee. We have to register. We have to apply in the website in NBPGR website. So the procedures will be dealt by the Mr. Rajiv Gambhir. So after 10 minutes, not 10 minutes, 8 minutes exactly. So please be online, and after a short break, that lecture will be continued. So thank you everyone. So be online. And it will be taken over by Dr. Rabi.
Okay, ma'am. Okay, just. Welcome, Gambhirji.
Thank you, Mr. Dr. Ravi.
गंभीर जी 3:30 बोला
Yes.
पहले पार्टिसिपेंट्स को ना, इसलिए वो लोग
No problem.
Already they are online ना, तो ठीक है, you can start I think by 2 minutes.
हाँ ठीक है।
नमस्कार सर, नमस्कार।
अच्छा गंभीर जी, एक बार मैं अपना शेयर करूंगा
जी,
जस्ट इंट्रोडक्शन के लिए। दिख रहे हैं ना?
Yeah.
Yes. Welcome back, dear participants. Let me introduce our current speaker. She is Mr. Rajiv Gambhirji. He is the Chief Technical Officer, AKMU, ICR, NBPGR, New Delhi. And he is having a vast experience of more than 30 years in web applications as well as IT infrastructure in my institute and other databases also. And he is a main key role in development of development and management activities of PGR databases. And he is well expertise in networking and LAN systems also. And he involved in 2 PI projects as a co-PI. He involved in 2 institutional projects. That is a prestigious PGR informatics and development and implementation of novel algorithms and software modules for PGR informatics. And I can say it is a proud, actually he is one of the member in development of our PGR portal. I think PGR portal is in India, it is the best one to access our passport data information and other things. And even the other databases also there, that is. And let me invite Gambhirji, welcome you on behalf of team and my organizing members. Please kindly share your experience and please mention how we can upload the details for trade specific registration and other data.
Yeah.
Please welcome Gambhirji.
Thank you, Dr. Ravi. Good afternoon, dear friends. In today's session, we will see practically how we can place an application for registration of germplasm material. I'm going to share my screen. I hope you can see the screen now.
No.
No?
No.
Is it okay?
It is okay, but it is not in PowerPoint mode. Just in PowerPoint option, you just click. Okay. It is okay.
Yeah.
Actually,
on this germplasm registration information system, you had a great detailed discussion on the 8th of this month that was given by Dr. Veena Gupta and supported by Mr. Arup. I think you have already seen a glimpse of that particular screen and some tidbits of the procedure, how you can apply online is already in your knowledge. First of all, we begin with that how to reach that particular system, that is GRIS. The most common way is to use this URL, http://www.nvpgr.org.in, that is through the NVPGR website, followed by port 8080 and then /registration. Another alternative is to go through PGR Informatics dashboard, where you can say that pgrinformatics.nvpgr.org.in, followed by web apps. All the web apps will be displayed before you, listed before you, out of which you can go to the particular germplasm registration. Or the most simplest way is to go to the NVPGR website, where on the left-hand side menu, you can find the option of germplasm registration. Just click on this. Either way, whichever option you use, that will take you to the home page of this particular application, where you will be prompted to log in if you have already registered yourself. And if you have not registered yourself, then you will have to sign up first of all. So sign up to this particular screen is necessary to place your application for registration. Now, before I dwell into the details of registration, I would like to have a 5 to 10 minutes discussion on the technical part, which in my opinion is almost necessary in today's scenario. This particular screen shows you that how any web application or an application which we process on internet works. In this example, we can see the same URL of our registration application, where a user like you and other users can place a request on the internet, and that request, after passing through a router or a firewall, reaches NVPGR server, where the server application GRIS is there in the form of an application and a database. And why I am showing you this particular screen? Because I know that each one of you is a subject matter specialist. You have your own area of specialization. But today, each and every professional related to any subject area cannot do anything without the IT, the information technology. Sooner or later, willingly or unwillingly, you will have to come across the IT related things, whether you need to get any application developed of your own, or suppose that if you want to develop a page of your own. Nowadays, everyone is familiar with simple files like PDF and doc and XLS, many other files, through which we usually communicate and send our data or information from one place to another place. Sooner or later, you will be required to develop or get developed any application of your own, because we received many queries about the same. And I just want to explain you this through one simple slide, that what particular questions you are asked and what we have used in this particular application of registration. As I am telling you that any web application comprises of two parts. One is the application software, the other is the database. The application software is of two types, either open source or the commercial. Open source are those which are available to you free of cost, freeware, requiring no license at all. And commercial ones are those which require you to pay certain license fee to acquire certain keys and then get your work done on that. Similarly, similar to the application software, databases are also of two types, the open source and commercial. There is only one technical slide that I'm showing you. After that, we will simply pass on to the registration process. And when you want to get any application developed of your own, then major question before us is that what particular application software we should use, what database we should use, whether we should go for open source or commercial, or what things are to be kept in mind. Now, I have listed a few common application softwares in which you can get your application developed. out of which the PHP and the Python, these two, and the Java, these three are free open source available in the market. Very easy to learn. Even school going children are using these, and you have to just spend a time of 1 week to 10 days to learn this, and you will be very confident and happy to learn that a simple application of your own you will be able to demonstrate related to your work on any platform. And sooner and as soon as you graduate through this simple application software development in PHP, Java, or HTML, then you can start thinking of how you can include the database also. This is only the technical part that I wanted to show you. In case if you have any queries on this particular technical part, whenever you are coming across your requirement of developing any application, you can simply throw a mail to us and we'll be very happy to help you out. Now, coming to our main application of
गंभीर जी, ये पावर पॉइंट मोड में अभी निकल गया।
मैं वो दूसरे एप्लीकेशन लोड कर रहा हूँ।
हाँ, ओके ओके ओके, हाँ ओके ओके, ठीक है।
जयेश शर्मा, ठीक है।
Can you see the screen now?
No, sir.
No. It's not coming on the screen.
Screen not yet shared.
इसको भेजना है। Is it visible now?
Yes, visible.
Okay. So the URL that I have informed you, that if you go to www.nbpgr.org.in, you will go to the home page of NBPGR website, from where you can follow the germplasm registration link, and you will be led to the home page where you will be prompted to sign up or log in. So if you just have not signed up, you can simply click on this sign up, and a simple form will be displayed before you, where you can fill in your basic profile information details, your name and username. Username will be of your own choice. You will have to use select the username of your own choice. Then the date of birth, gender, date of birth, designation, division, and all the information which is preceded by an asterisk. It is mandatory to fill. Almost each and every field on this page is mandatory for you to fill in. Now, once you fill in this form and click on the submit, this information will be sent to our server, and you will receive an email on the email address that is given by you in this form, and a registration will be done. On this email ID, you will be receiving your registration information along with the password. The password which will be generated by the server will be quite cumbersome for you to learn. So what you can do is, suppose that I'm taking you to this form, and on this username, the first time username that you have given, in my case, it is this. The first time user ID and password will take you to the home page. And in any case, if you want to change anything in your profile or the password, you can simply go to the settings. There are the two options of changing the password or managing your user profile are available. If you need to change a profile, you go to there and your profile detail will be there. You can do whatever changes you want. And if you need to change the password, you can simply click on this. Here, the old password will be the one that is just sent to you through the server. And in turn, you can select any new password. And this new password, remember, should be quite easier for you to learn, but not as easy to be assumed by the other people. So once you will enter the password, then you have to retype the password and confirm the password, then submit. Your new password will be stored on our server for future use. And remember, in case you forget your password at any stage, you will have to remember your username only. You just enter your username and click on this forgot password name or password. Now here the email ID that was given by you at the time of registering yourself is the most important detail, followed by your date of birth. Combined on these two, you will be getting an email reply along with your user ID and the password. And again, the same process you can use to change your password if you need to be. Now, proceeding further and coming to the actual business of placing an application for registration. Once you will log in into the system, you will be getting these 5 boxes. These 5 boxes are basically to keep a record of your activity on the germplasm registration application. Submitted applications currently is 0 and all other things are also 0. The incomplete application is 0. The application for revision is 0. Whenever you will apply for a new, you will place a new application, you will come into submitted application will become 1. Now I will try to make a new application for you here. Whenever you need to start entering your application, you need to click on this germplasm registration. This is a dashboard. Remember, this is a dashboard showing you your activity details on registration application. If you want to start actual application, you have to click on germplasm registration. Now this germplasm registration has again 3 activities. One is new application, second is incomplete application, third is revised application. So I click on this new application. Each new application will take 6 pages of information from you: primary information, developer information, passport and pedigree information, remarks, attachments if any, and undertakings for final submission. And at the bottom of each page, you will find an all-important click, that is save and continue. Once you see a page, you go through this and fill in the details carefully. And after each data filling, you have to click on this save and continue in order to proceed to the next page. I'm repeating this because it is very important. Your complete registration application will be involving 6 pages of information, starting with primary information. Whenever you will fill in the details of one page, in order to proceed to the next page, you have to click on this save and continue. Simply clicking on developer or on any of the page title will not take you to that particular page. What I have done is I have downloaded an application for you to see, and let us see how to apply. I want to apply for Phaseolus vulgaris. Now remember that whenever you will start entering the botanical name, you will have to choose the one which is there in the drop-down list. Phaseolus vulgaris. The detail about the crop name, the common crop name, the crop group name, this will automatically be displayed to you. And because this was a new application, the application status new will automatically be displayed and you will not be allowed to do any changes here. Now, what is the biological status of the material to be registered? It is a germplasm collection, it is a genetic stock, recombinant, mutant, or any other. So whenever you will say any other in any of the options on any page, if you will go for the option others, you will be asked to describe that what others do, what do you mean by others? So you will have to be very crispy in giving the details about the others, or you will have to select from the pre-filled options like this germplasm collection. Nature of the genetic material is seed. It may be seed, tubers, roots, bulbs, and whatever it is. Now here, in the last session, I got a chance to hear few of your queries. There were queries you asked that how many seeds we need to present at the time of making an application, and whether IC numbers is important for applying for registration or not. We will be handling these types of queries also in this session. Now, after entering this nature of genetic material as seed, now the identity of the material. What is the identity? It may be the national identity, or it may be any collector ID, it may be a variety name. So whatever identity by which your germplasm material is to be identified, that you have to select from here, or you have to type. You have to simply type it from here. Quantity deposited. This is again a necessary field. Number of seeds or clones that is entered by you, that is submitted by you. It is 500. Now the criteria of germplasm unique features, this is maximum 3. Here you have to be very careful because you have to give the most important unique features that you need the committee to take into account at the time of considering your proposal for registration. In my case, let us say that it is resistant against white mold disease, and this may be a single trait or there may be 3, but at the most you can give 3 traits only. What is the material value? Is it a scientific, commercial, or academic, or other? It is scientific. And again, if you will select other, you will be prompted to describe that what do you mean by other. Basis of eligibility, published with peer review, all India coordinated trials, and again, any other. Has it been registered or protected anywhere? If you will say yes, obviously it will be rejected because the material already registered somewhere else. The application for re-registration will not be considered. So only new application where the material is not registered or protected anywhere else will be considered. So this is the first page information, which in my opinion is complete for my case. So I simply click on this, save and continue. Now comes the second page of developer. Here, 2 parts information is to be given. One is the name and address of the corresponding person. The other contains the particulars of the scientists or the persons or the organizations, farmers, farming community, whatever, who are involved as a group in the development of this germplasm or the genetic stock. The person who is a developer may also be, the person who is in the corresponding person may also be a part of the developing team. Now, suppose that if this is the case, we say that Dr. Mohar Singh is the person who needs to correspond with principal scientist. And mind you that each and every detail is followed by an asterisk. So this simply means that this is compulsory. NDPGR regional station Shimla. Telephone number is not necessary. Fax is not necessary. Gmail or mail is necessary. Here I am giving my own mail. And then the mobile number. Again, I am giving my own mobile number. Just we are having a trial testing application here. Now, suppose that the person, Dr. Mohar Singh, is the developer also. So this information needs to be entered here also. So the one mistake that generally people do is that the first developer they write over here and they forget to mention the developer again here in the second part. This part, the person who is the corresponding person may be or may not be a developer. So that is why we have to make such a provision that the person who is a corresponding person, if he is the developer also, this information should be repeated over here. Mohar Singh, principal scientist, NDPGR regional station Shimla. And now, if you want to add more developers, there is no limit that how many developers may be there. There may be one single person. It may be 2, 3, 4, 5, any number of 10 people may be involved, 20 people may be involved. So you have to simply keep on adding the person's details over here. and proceed on. And once you are through this detail of corresponding person and the developers, again you have to click on this save and continue. At any stage, if you have proceeded to the page 2, 3, 4, 5, and at any stage you need to go to the previous page, the option appears here to move to the previous page. Otherwise, to proceed further, you have to simply click on save and continue. The third page contains the information about the passport and the pedigree information. The first detail asked from you is the national identity, that is IC or EC, and the question that I remember very much on that day raised by 2-3 people rather, that whether receiving a national identity IC number before making an application for registration is necessary or not. I am answering the question to this reply in 2 parts. Number 1, if you are going to place a new registration request, mind you, registration request, the IC number necessity is not there. You do not require to beforehand acquire an IC number if you are going to place an application, new application for registration of germplasm material. But in any case, the second part that I'm going to tell you, the germplasm will be registered only with an IC number. So in a case, if you will priorly receive an IC number, you can mention it over here. The same will reflect in your application as well as the same will reflect in your registration certificate provided to you after proper recommendation of the committee. And if you do not acquire the IC number beforehand, at this very moment also you can make a request that I do not have the IC number right now. I need to apply for the IC number also. In that case, your request will be accepted once you will say the option no. So if IC number is not beforehand received by you, a simple form will be displayed before you containing all the necessary details that we need to allocate any IC number. Here the necessary detail is the collector number or the collector ID, any other ID. This is or either this or this. The source where from, it may be a farmer's field, it may be an institute, it may be a farm store or whatever the source where from you have collected it. The village, the mandal, the district, the state. And if it is an institute material, you can give the address of the institute itself. State is necessary. And once you fill in this detail and save it, this detail will be sent to us. And before your application for registration of material is processed, IC number will be allocated automatically by ourselves. So it simply means that your application for registration of germplasm material will end up automatically with an IC number. So whether you receive it beforehand in advance or you apply it from here, IC number in any case will be allocated. Without national identity, mind you that your application will not be considered by the expert committee. Now, if you have already got an IC number for the material, like in my case, it is 278744. Is not taking the type from here. No problem. I can place a new request. It is new germplasm. Source is farmer's field. Let us see. So this detail will be saved over here without the IC number and it will be sent to us. Now, if there is any farmers or community contribution, you can simply type it down. This is simply a text box where you can type anything. And obviously you will have to keep in mind the maximum limit of 500 characters. And Proceeding further, you have to give the pedigree of the genetic stock. If it is genetic stock, you have to give the pedigree. Breeding method is any one of these: introduction selection, mass selection, pedigree selection, pure line selection, and so on. And again, for others, you will be asked immediately to describe what others means. If it is pedigree selection, and let us give some sample, like this is the pedigree. This part is over. Any other information related to the usefulness of the identified germplasm or the genetic stock, if any you want to give, you can scribble it down in this particular text box. Now, the year of production, year of seed production is again compulsory. Let us say it is 2016. And season, let us say it is Rabi, or any season maybe. The location of the seed germplasm is, say, MBPGR, Station Shimla. Quantity of seed available is whatever it is, let us say 20,000, 10,000, 1,000. Seed viability is this. Now there may be a question that if a particular crop group is there, then what is the minimum seed viability which is accepted? Most of these questions, you need not send any email, but these will be available within this application. Only once I will finish this data entry part, I will show you that where you can go and you can cater to most of your queries. The seed deposited, whether the seed has been deposited with the National Gene Bank, yes or no? Yes or no? If it is no, then where the material is available, where is, at which NAGS, at which, if it is in Field Gene Bank, in which institute the material is available. And if it is simply yes, it simply means that you have deposited the material to the NBPGR National Gene Bank. And after finishing the information on this third page of passport and pedigree information, you can simply again click to proceed to the next page, save and continue. It's not taking. We have fed all the necessary information here. A little hitch it is creating since last 2-3 days, but in any case, if the first 1, 2, 3 pages of information you have entered, you can simply go to the home, and at any stage, it is not necessary that you fill in the complete 6 pages in one go. You can enter 1 page of information and you can leave the incomplete information in between and go away from your system. But you have to keep remember that whenever you want to re-continue with the new application that you previously left incomplete, once you log in into the system, on the dashboard itself, it will show you that you have one incomplete application in your bag. What people mostly make a mistake many times is that they every time start making a new application request, ending with multiple duplicate requests, which are of no use for us, rather it creates a problem. However, we made a provision to restrict such duplicity, but that also generated problems for many applicants. So we had to remove that restriction. But now you have to remember only the thing is that once you have started making any application, and if you have partly filled it 1 or 2 or 3 pages, then your status will be there in the incomplete application, where after just clicking on this, you can see that the application is there. But again, I am telling you that whatever action you need to take on your application, for that you have to click on this germplasm registration. There are 2 things. One is the dashboard, that is to view only. There is a homepage of your the application processing, where you will be led to by this germplasm registration. Now here, your incomplete application will be found in the second option of this incomplete application. Here, the action will be viewed by a pen or a pencil, clicking on which will allow you to continue further where you left last time. Now save and continue to the second page, save and continue to the third page, and here the detail 2787, 2787. रत्नेश ये टाइपिंग नहीं ले रहा है यहाँ पे देखो रत्नेश seven 8744. So after entering this IC number, automatically the details have been entered. Farmer's contribution, if any, no. Breeding pedigree method is there, and this is the detail that I have entered. Year of production, again I am repeating the same thing because this was not saved last time. 2018. Unused. Unused. Other information mandatory. Okay. So season of seed production is Rabi. Location of seed production is NBPGR Regional Station Shimla. Quantity of seed available is 5,000. Viability is 95%. Seed deposited in nitrogen bank, yes. The usefulness of the identified germplasm of the seed stock is compulsory. So we can enter some detailed information like this over here. I am copying and pasting it from another source to save my time. So once this detail is entered in the passport and pedigree, we can again save and continue. Click on the add button on the passport. Okay. Right, like that. This. You have to just click on this add button to add the national identity details over here. And once it is added, now you can go to the save and continue. Now the first 3 pages of registration application are over. And now we have taken to the remarks. On this particular page, the 2 most important things that you have to consider are, the first is the manuscript, the other one is the recommendation of the Institute's Germplasm Identification Committee. Both are associated with asterisk, means both are necessary for you to click and upload the detail. And the most important thing here to keep in mind is that you can make attachments in the form of PDF files only, by remembering that the maximum size of the file that you can upload is 5 MB only. You have to just click on this choose file and you will be taken to your computer's hard disk drive or the source where from you can attach a particular file. I can take any sample from here, let us say like this, and let us say open. And once this is done, then you can go to the other file, choose recommendation of the institute. Let us say this is the recommendation. Let us say okay. The only thing practically you need to remember is that the files you are uploading, trying to upload here for manuscript as well as the recommendation of Institute Germplasm Identification Committee should be PDFs only, should be within the limit of 5 MB of size. And once you have done this, and if you have any other details which are not covered as a part of these files which are uploaded by you, with the salient features and the chief botanical and morphological agronomic description, you can simply type it over here. And any other detailed information which you feel is important for consideration of the expert committee, that also you can type it over here. The necessary things are these 2 uploads. And once these 2 uploads are there, you can simply click on this save and continue, and you will be taken to the 5th page of attachments. Now here, what kinds of attachments are required to be given? Remember on the first page, what you did was the material value is scientific and the basis of eligibility is published in a peer review, or it is an all India coordinated trials data, or it is an institute annual report. So any attachments related to these basis of eligibility, these you can now upload in these attachments. And see how I am moving through the pages. It is only a single way of clicking onto this save and continue. Now attachment of the file. Now here the attachment is requiring your simple consideration. First you have to give a title of your own. Title of your own. You have to type a title of your own, that what attachment you are going to enter is what? It is trial data, or is it a research paper, or is this an annual report? This. So that we should come to know that whatever attachment you have given over here is what. Then you have to click on this choose file. Now in this case, if I say that I have chosen this particular file and say open, now this file name will be displayed over here. The most important point here to consider is that after choosing the file, never hope that it is already uploaded. It will be uploaded only when you will click on this upload button. So once you will click on this upload button, this file will be reflected in the portion on the second part of the page. Now you can click again to choose a file of your requirement. It may be a research paper, so you can keep on adding any number of attachments by giving a proper title and clicking on the choose file, followed by the upload. And each time you will select a file and upload it, it will be listed before you in the part beneath the selection part. And once it is done, you will be sure that the item has been successfully uploaded by you. Suppose that in this case, I take one more upload, and this time I have uploaded a research paper to substantiate my claim where the information has been published, and then say upload. The research paper is also uploaded over here. Now suppose that if you want to see that whether you have uploaded the correct document or not, you can simply click on this. It will be opened before you, and you can see the contents and be assured that the thing Control X. So you can simply click on the file names or the titles which you have just uploaded to see that whether you have uploaded the correct document or not. And once you are through that, yes, the correct documents have also been uploaded. So 95% of the work is over. Primary information filled by you, developer details, by remembering that the person who was the corresponding person, whether he has been included in the developer or also if he is the developer or not. The passport and the pedigree information, remembering that the IC number details, whether it is already received by you or not, the remarks and the attachments finally to substantiate where the details have been published. Once again, clicking onto the save and continue takes you to the final page of undertaking and submit. Now here, only thing that you have to give is your response to these radio buttons, to these 14 points, just to ensure that your information, that your details of the new application have been filled by you completely and nothing has been left to discard your application. Whether this application is new, if you will say yes, obviously automatically the revised application will become no. Whether the same or similar material has been registered earlier is no. Whether unique or distinguished characteristics have been entered by you? Yes. Whether documentary evidence has been provided by you? Yes. State any other economic potential value of this is possible? Yes. Whether applicant, institution, university, and center has given the commitment for maintenance of the supply of the germplasm to use? Yes. Whether appropriate size of germplasm sample for long-term storage has been provided to NGBS? Yes. Almost these questions will end up with yes. And once you have filled in this checklist, finally, you have clicked on this checklist, you have to just agree to all the terms and conditions, which is an obvious part of any new application anywhere. And you can simply say preview and submit. Once you will click on this preview and submit, it will show you the complete preview of your complete information, that details you have just entered in. That is, this is an application for registration of Phaseolus vulgaris, bearing identity number this, corresponding person is this, the developer or developer's details, followed by the passport details of the information, and then the pedigree and all other things. Now the manuscript and the recommendations of the institution germplasm identification committee. These two you have just said that yes, these are there. So if you will just click on this, be assured that you have entered the correct information. This is the manuscript carrying the crisp information that you want to bring into the notice of the expert committee to be considered. And this is the institute's approval. Generally, the institute's approval is a part of the hard copy manuscript that you are required to send at the end of filling this online application. So what you can do is you can simply photocopy it from there or scan it, make a PDF of the same, and then attach it here along with your application. And once you are satisfied that, okay, the details that have been entered by me and the attachments are okay, then you can simply go for the final submit. Before you click on the final submit, I would like to draw your attention to the point that once you will finally submit it, you will not be able to make any changes. If you just after 5 minutes or 10 minutes, you come to know that, oh, this information was left, or this information was not given by me, or this wrong information has been entered. So be very careful at the time of clicking on this final submit, because once it is submitted, no modifications or editings will be allowed to be done later on. And if you still want to go into detail that what attachments I have entered, what is the application checklist. So till this stage, you do not click this final submit. You can go through this application as many times and you can make changes as many times. And once you are through that, okay, now this is the time that I have seen it very carefully and it is okay, you can go to this final submit. Again, there is something blinking on the screen in the red color. You have to go through this very carefully again, because this is a mandatory part. After final submission of this registration application online, you have to send 2 sets of hard copy of the application along with the documentary evidence duly signed by the associates, depositors, and the head of the institute. The copy that I have just shown you here is the recommendation of the institute's germplasm identification committee. It is recommended by the authority, competent authority, and it is also signed by the partners. So now I am going to finally submit my application. Now it is asking you to check. It is just a cautionary. It is just a cautionary message that once you submit it, you will not be allowed to do any changes. I am sure. So I am proceeding further to okay. Now, once you finally submit it, on the very same page, the save part, the final submit part will be, the picture will be converted into print. You can simply go to the print, click and take a hard copy printout, or even you can send it to a printer, or you can save it as a PDF also. So by this, your exercise of applying for the registration for germplasm material will be complete. So once it is done, you can always go to the home page to see that the application from incomplete has been shifted to submitted application. Now this application will be considered and further forwarded for the consideration of the expert committee. They may recommend your application in one go if it satisfied to the complete criteria, or they make certain comments and send it back to you for revision. So if this particular application comes back to you for any revision, it will be reflected over here. Instead of 0, it will become 1. After clicking on this, you can open it again and satisfy to the queries, whatever have been raised by the expert committee. You can resubmit it, and again it will come back to the submitted application. And this exercise will go on till your application is not finally either recommended or not recommended. If it is recommended, it will be shown over here. If it is not recommended, it will be shown over here. Obviously, the not recommended will be with some reasons, with certain points clearly mentioning you that why your application has been rejected. Now, coming to your general queries, from whatever we have done till now, this was done only from this tab of home. Other than this, there is about us, which is for you to go on and read. But the other things for your interest will be the announcements in the proceedings of the previous committees. As you can see that this is 43rd PGRC committee minutes which are available for you. Actually, it is I think 48th or 49th meeting which has taken place last time. If you just click on any of the meetings, you will be given the complete details of the committee constitution, the number of applications considered, whether these have been recommended or not, and if these have been recommended, then what was the INGR, Institute, the germplasm registration number that was allocated to this, that will be listed before you. So this is the outcome of the minutes of the 43rd Plant Germplasm Registration Committee. So similarly here, if you click on view all, you can see that for all the PGRC committees, the minutes of the meetings are available over here. The most important tabs on this home screen for you are the guidelines and the help. If you click on this guidelines, whatever questions are raised in your mind with regard to the procedure of registration of germplasm will be answered over here. You will find answers to your 90 to 95% queries into these guidelines. These have been prepared so and elucidated so easily, covering into detail all your requirements. Like the last time there was a question that for orthodox seeds, how many seeds we need to present. So you can go through this, how the screening of the application will be done, what will be the validity of the registration, and if it can be deregistered or not at all. Then the guidelines for submission of the recalcitrant intermediate seed germplasm. At least 200 to 500 seeds should be supplied. So all general queries will be answered there. For orthodox, like Dr. Veena Gupta informed you last time, that for the self-pollinated it is 2000 and for cross-pollinated it is 4000. That also is answered within this particular manual only. And what are the national active germplasm sites where the material is available? The complete names and addresses and details are also available. So I simply mean to say that the procedural queries will be answered in the document which is available in the guidelines. Whatever functional means functional with respect to this particular application. कि कैसे सेव करना है, कैसे आगे जाना है, which part is necessary, how to proceed further. For that you have another option of going to the help. This help will take you to a presentation of the user guide of this complete application, how you can register yourself, what is the procedure of changing or entering your profile, how you will be given the username and the password. I told you that the system generated password at the very first stage will be quite cumbersome for you to learn, like this, which you can change always from going to the home and settings. Home and settings. And within this, further, this is the dashboard showing you that after registration, all these boxes will be empty, showing you 0 because you have not yet entered any single application. And then proceeding one by one, whatever I have just discussed with you in detail, page wise, how the information is to be entered. The only thing is that you need to just start entering your information after clicking on this Jira Plaza registration and new application. I'm just having a revisit on whatever we have discussed right just before. 6 pages of detail. 1 page after another, you have to fill in the details. All the details which are associated with an asterisk are necessary to enter. You have to click on the save and continue each time you want to proceed further to the developer information. And every time you fill in the application partly, 1 or 2 or 3 pages, the incomplete application will be shown over here. Never ever create a new application for the same material if you already have created an incomplete application in your application in the box over here. So every time you give a new application call, you just have a glance that whether you have any incomplete application related to this very same work or not. Okay. Then finally, once something is available, you can always go to the germplasm reports, and this germplasm reports will show you the reports of all your own individual applications. How many new applications I have formed, how many of my applications are there approved ones, how many are there not approved ones, how many are there in revised ones, and what are their details. Right now, because I have just created a new application, if I click on this and submit, it will show me that this is a new application that is lying pending with the NBPGR for consideration of registration. So this is the. Now suppose that if after finally submitting your application, where you yourself are not allowed to do any changes online, and suddenly you come up with some information that, oh, this requires some change. What to do in that case? Or if the query, whichever is entered by you, whichever is bothering you, is not answered anywhere in this guidelines manuscript. In that case, you can always go to the contact us. In the contact us, the 2 email IDs should be of utmost importance to you. First one is that anjali.call@icr.gov.in. The person who is responsible for clearing all your doubts related to the procedural queries, how to apply. Means all the procedural queries which are covered under this guidelines. And if any application related queries are there, you are not able to change your password, you are not able to make any changes in the profile, or you are not able to proceed further to after finishing the details of the first page, the second page, or the third page, or so on. These types of queries you have to simply send to akmu.nbpgr@icr.gov.in. Now here, I want to emphasize that suppose that if you have made an application for allocation of IC numbers to NBPGR and your request has been delayed for some reasons, and you want to inquire that whether IC number to my material has been allocated or not, means all IC number related queries also you need to send to acmud@nvpg@icr.gov. Okay, I hope this is clear. So your procedure, germplasm registration information system related procedures queries you need to send to anjali.com call at ICR, and IC number related and application related queries you need to send to acmu.nvpg@icr.gov. Now here, for your convenience, another tab is available to show the inventory. So this inventory shows you that how many germplasm are there which have been registered till date. The number is well beyond 2000. It is, I think, 2064 or 74, but it is showing you 1733. And why it is so, and I will just tell you why it is so. Here you want to see that what are the cases which have already been approved for registration related to the rice field. So see carefully. What I have done is I have started typing the crop name over here. Simply typing and clicking show will not help me. I will have to just wait for a while to select from the drop down list the crop of my choice, and then clicking on the show will show me the output, where it is showing me that there are total 2 applications related to rice bean which have been given the INGR number, which simply have been recommended for registration. And this medal sort of icon is just to show you the QR code. And if you click on this, all this detail, the further details for which you will be shown in QR. And this QR code you can simply scan on any of the apps on your mobile. It may be a Paytm or it may be any other QR code scanning app. And after scanning this, the details of this particular application will be simply displayed on your mobile itself. Now suppose that if I say it is rice and I want to further squeeze my search, and there are so many, there are 176 applications of rice, and my application for rice is also there, but I am finding it quite time consuming and difficult to see that out of 176, my particular application was related to ARC 10659. So what I know is the crop name along with the donor identity. Somewhere some part was ARC or it is 10659. So combining these two or multiple information on this form will narrow down my search. And after clicking on this, I see. Sure. अभी हम इसको ऐसे करते हैं, बासमती ले लेते हैं पूरा. I click on this basmati 370. So including these 2 parts in my criteria and clicking on this show. So in any case, if you the purpose of this form is to narrow down your search. If you have already made an application, you want to see what is the status and what is the final status of my application. So from here you can see that all the recommended details, all the materials which have been recommended for registration will be listed before you, and you can go through one by one. Now coming to the question of this 1733 only, and whereas I am telling you that almost 2074 applications material have been registered. इसको कैसे करें नीचे यार? Actually, the thing is, if you just, I am able to demonstrate it to you. If you just see on this URL over here, nbpgr.earthnet.in/8080. In reality, the application is accessible by typing registration over here. Registration after 8080/it is registration. For the sake of convenience of the applicants or for the trainees, we have created a clone of this application where it is shown as www.nbpgr.earthnet.in/8080 followed by /GRIS training. If you will follow this path, instead of registration, if you just mention GRIS training over here, it will take you the same type of application. Everything will be same, ditto. The only purpose is that in order to make any rough applications where you do not want your applications to be considered at all, you can use this URL www.nbpgr.earthnet.in/GRIS_training. Whatever applications you will create over here will be considered just for the sake of training and testing. Otherwise, the procedure will remain the same. And if you want to make an actual application for consideration for registration, then you have to simply go to our website www.nbpgr.earthnet.in followed by the link of germplasm registration, and the same application will be accessible from there also. So I hope that I have been able to completely demonstrate this application to you. So now if you have any functional queries, I am emphasizing that functional queries because we are totally IT people. So if there are any specific procedural queries, which if I am able to, I will be happy to reply to them. Otherwise, for your procedural queries, you will have to contact someone in the germplasm conservation division, like Dr. Angelika. And for this application, if you have any queries, I will be glad to handle the same.
Thank you, Gambhirji.
Thank you.
Dear participants, if you have any doubts or queries, please can you ask, or otherwise you can post in chat box. Actually, one question from Dr. Suma Mogali.
Yeah.
If
we have a AICRP data for my claims, is the recommendation of IGIC report mandatory? Again, I will say that this query can be answered only by the GCD, by Dr. Angelika or by the head of the GCD. But so far as I can reply that yes, the recommendation of IGIC is mandatory.
Mandatory, yes, sir.
It is mandatory because you need to upload the IGC recommendations along with the application and send it in your hard copy document also.
It is mandatory, sir. It is. Please, dear participants, switch on our videos. And you can interact if you have any doubts or queries.
Now, I am repeating once again with which I began. Many scientists, many subjects, material specialists come to us with their general queries के साथ this application मेरे को develop करना है, बताइए किसमें करवाऊं और मुझे खुद करना है तो मैं किसमें करूं. और मैं आपको बता रहा हूं कि in today's scenario, having knowledge of IT means the web application development or some sort of application development will be an added advantage to each one of us. And what I can suggest you is to begin with the mother of all languages, which is understood by each and every web browser. And this is simply known as HTML. You just go back to your home and you ask your school-going children that what HTML is. They will tell you what HTML is. And it is very, very easy, English-like of language, using which you can draw a simple web page of your own. And you can see that what happiness you will get that once you will develop a web page of your own. I will suggest you to begin an HTML page of your own just by displaying your own name, a single line information. Like you create a web document, like you create a Word document having an extension of .docx. The HTML documents have an extension of .html. and just see how you can display your name on HTML. And once you are through with this HTML, and I can assure you that if you will develop your interest, it will not take you more than a week or 10 days time to learn HTML and to develop your complete biodata. Because we come across many people who come to us with the solutions. They ask us, sir, वहाँ पर हमें biodata भेजना है, and they are not accepting PDF. They say that you should have your own page. When they say that you should have your own page, it simply means that you should have your own HTML page. And creating an HTML page, I am telling you, is a child's play. It is a child's play. This is one scenario. Or the other scenario is that some fine day, you may come across a situation where you need to get a software developed from some outside agency, from a third party. What kind of discussion you should have with these people? कि कैसे हमें अपना एप्लीकेशन डेवलप, आपको अपना रिक्वायरमेंट तो मालूम है कि हमें अपना प्रोजेक्ट का ये रिक्वायरमेंट है, हमें अपने डेटा का ये रिक्वायरमेंट है, तो आपको जो चीजें उनसे डिस्कस करनी पड़ती हैं कि हमें फ्रीवेयर यूज करना है कि शेयरवेयर यूज करना है, पैसे बचाने हैं तो हमें फ्रीवेयर यूज करना है, अगर हमारे इंस्टीट्यूट के अंदर ऑलरेडी कमर्शियल सॉफ्टवेयर अवेलेबल हैं तो हमें कौन से कमर्शियल सॉफ्टवेयर अवेलेबल है यूज करने हैं, फ्रीवेयर को यूज करने के लिए आपका एप्लीकेशन सस्ते में डेवलप हो जाएगा, रिक्वायरिंग लेसर मनी, और अगर आपको कमर्शियल एप्लीकेशन कमर्शियल सॉफ्टवेयर यूज करेंगे तो उसमें आपका पैसा भी ज्यादा लगेगा, रिस्ट्रिक्शंस भी ज्यादा लगेंगी, और हमेशा लाइसेंसिंग का और जो आपका जेन्युइन सॉफ्टवेयर यूज करने का जो आपको कंपल्शन है वो हमेशा आपके साथ रहेगा। मैं आपको श्योरिटी दे रहा हूँ इस बात की कि आप आज अगर आप सोचें कि अभी हमें इसकी जरूरत नहीं है, आज से 5 साल के बाद, 8 साल के बाद, 10 साल के बाद आपको सुनने को मिलेगा कि मुझे एक एप्लीकेशन बनाना है PHP में, मुझे एक एप्लीकेशन बनाना है HTML में, and your school going children will be able to do so. But what we expect from you is just to know the terminology of these things. We don't want you to be expert of these things. ये IT क्या है, HTML क्या है, PHP क्या है, Java क्या है, ठीक है ना? And the best teacher each one of us knows is Google. So I finished my talk with this over here. And if you in future have any query, you simply throw it to us on akmu.nbpgr.icr.gov.in. And the queries are welcome even from whatever I am suggesting you, that how to learn this HTML, how to learn other things, or anything related to the general present material registry. Thank you very much.
Thank you, Gambhirji. And there is no doubts from participants, I hope. Actually,
doubts will be generated
doubts
only
will
once,
be yes.
Only once they will start laying their hands on the keyboard, they will try the application, only then the doubts will generate. And we are open every time. We just you simply send your query to us on email and you will get a proper reply, or you can talk to us on the phone also. But we want you to use this. Yes. And the most important thing is that one lecture you got from Dr. Sunil Archak on PGR informatics also. In this pgrinformatics.nvpgr.earnet.in, you will find mention of all the applications which are there. IC number related, EC number related, gene bank related, whatever material is available in gene bank. So I will suggest you to go to this pgrinformatics.nvpgr.earnet.in. The complete list of all the applications developed by NVPGR are there. And these are very user-friendly, very simple applications. And I hope that once you will go through these applications, you will find something important and useful to you. Okay, thank you.
Thank you, Gambhirji. On behalf of organizing team and other team members from participants also, I am really thankful to you for briefly.
Thanks to everyone listening to me so patiently, and thanks, Dr. Ravi.
Deeply explaining explaining all the things. Actually, this is very important. Actually.
Yes, it is.
Lot of people don't know. Even me also. I also today I sat.
I'm sure that once they will start using it, they will be more and more interested in using this.
Yes, yes, sir. Thank you very much, Gambhirji. Thank you very much. Thank you. And today it is a concluding time, and tomorrow we will meet at 9:30. We have one good lecture by Dr. J.C. Rana, that is a mainstreaming agrobiodiversity for livelihood analysis and how to control the climate change vulnerability. and tomorrow we will meet again. And thank you very much, and have a nice day. Thank you, Vijay Mandalji. Thank you very much.
Thank you. बंद कर दूं?
हाँ, बंद कर दीजिए. Thank you. Okay, bye.
Bye.